Good morning and welcome to Life Beyond the Basics 2024. This is our PCLA's annual educational conference. I'm LoSasso, president of Parkinson's Community Los Angeles. And this year's event is unique. We are really excited about this because today we join together both in person and virtually from all over the world. 2020 was difficult, if not impossible, to gather. We quickly realized that this reality is leading to more loneliness, hopelessness, and loneliness. And so in March of 2020, we launched Let's Talk Parkinson's, a live online educational series designed to bring our community together. This initiative allowed us to continue learning, engaging, and fighting back against Parkinson's side by side. We also transitioned our support groups to virtual meetings, ensuring that our members could still connect, share stories, and offer support, all while fostering a sense of camaraderie and compassion. We now welcome into our community families living with Parkinson's from all over the globe, and all of our educational events we provide, like today, are free. Today is a celebration of PCLA's mission, our thriving mission, to empower lives impacted by Parkinson's by providing resources, strategies, and cultivating relationships. So welcome to all of us gathered here today at the beautiful Hotel Indigo in Los Angeles, and welcome for all our new friends from all over the world joining virtually. Welcome, welcome, welcome. We are dedicated to presenting the most current and inclusive information to help you fortify your PD toolkit. We strive to help you apply practical strategies for living and cultivate meaningful, practical strategies for living and cultural meaningful connections within the PD community. And this is what makes PCLA unique. We'd like to thank our partners who've made today possible: Abbott, Acadia, Boston Scientific, Medtronic, and Supernus. And if you haven't visited their tables, please stop by and see what new treatment options they have available today and what's coming down the pipeline. And if you're joining us via Zoom, we've posted links to their websites into the chat, and you can find out what they have to offer there. I'd like to thank our hard-working PCLA board and our community engagement director, Natalie Holtzman. Where's Natalie? She's probably working outside, scrambling, getting things ready. And our information and referral specialist, Charles Shield. Charles, thank you. You're also dedicated and amazing. We also have some incredible support group leaders who are here who have been with us since the beginning, so thank you. Nessa and Mike Weinman. Charles Coverdale as well is here, who's a support group leader, so thank you, everybody. PD is a progressive neurological disease that affects not just the individual, but indeed the entire family. But by creating a holistic and comprehensive approach to managing the Parkinson's journey, we strive to solidify a vision of hope, cultivate joy, and make lives better. This is PCLA, Parkinson's Community Los Angeles. We have wonderful plans for 2025, including more fun in-person events throughout the greater Los Angeles area, and new support groups and new online programming to help you live your absolute best life. So we're excited for today's program. We've gathered 5 experts from our medical advisory board who will be presenting some of the hottest topics of the day. But to keep you feeling invigorated, we have a fun and interactive movement session by Emily Ramon and Mackenzie McCoy from Reactive Physical Therapy and Wellness. They're awesome. It's going to be great. We also have an inspiring Mediflix short film, Stories from Living with Parkinson's, that we'll be playing shortly. There will be some time for Q&A after each presentation, and we will do our best to get to all of your questions. We are recording today's presentation, and it will be posted to PCLA's YouTube channel, which we are extremely proud of, and it has a wealth of information. You should check it out if you haven't seen our YouTube channel yet, but you will receive a link to today as soon as it is available. I want to again extend our gratitude to our partners, and of course thank you to all attending here and virtually. We hope our time together serves to be informative, dynamic, joyful, and inspiring. Now I'd like to introduce our first speaker, Dr. Wael El-Nachef, whose presentation is Gut-Brain Connection: Insights into the Microbiome's Role in Parkinson's Disease. Please give a warm PCLA welcome to Dr. El-Nachef.
Good morning, everybody. I really appreciate the host for inviting me to speak. This is my first time speaking in person in the PCLA, so this is a great experience. It's great to see everyone's faces. So I'm actually a gastroenterologist, but I have a practice that focuses on people living with Parkinson's who have GI issues. And so today I'm going to be mostly talking about the microbiome and some of the updates in research related to that and Parkinson's disease. We'll focus mostly on that, and then also I'll leave a lot of time for questions. If we have time, we can also talk about the general clinical issues we see related to this field. So yeah, in my experience, I've seen a lot of the GI complications related to Parkinson's disease. And a lot of these are related to something called dysmotility, meaning that the movement of the GI tract is not normal. And this can affect essentially every part of the GI tract. And so the GI tract is esophagus all the way to the anus, so everything in between. And so you can have issues with your esophagus and problems with swallowing. Your stomach can move slowly, and you could have gastroparesis. Your small intestine can move slowly. You get small intestinal bacterial overgrowth. Of course, there's constipation. And then if there's an issue with the reflex for defecation, you can get dysenergic defecation. Now, what's interesting about Parkinson's is that many patients have GI symptoms years before they develop the motor symptoms, before they get diagnosed with Parkinson's. In some cases, decades before. We'll take a step back and say, okay, what do we know is causing the problems in Parkinson's? Well, a lot of us are familiar with Lewy bodies, which are made up of a protein called alpha-synuclein. We pay a lot of attention to its involvement in the brain, in the nerve cells of the brain. And there's an area called the substantia nigra, the dopaminergic neurons there. And you can see that black stripe in that section of the brain there. That's what it normally looks like. When you lose those cells, like for example in Parkinson's, you lose that black stripe. However, the GI tract has a lot of nerve cells. In fact, we call it our second brain because it has as many nerve cells as our spinal cord. And there's a lot of thoughts that maybe this issue with the Lewy bodies can be found in the gut as well. And that might be what's playing out when you have GI symptoms. And not only is this a huge source of nerve cells in your gut, there's a lot of interplay between your central nervous system, your brain, and the gut back and forth. So these things work together. Now, I told you that the gut is the second largest nervous system in your body, but it's also the largest compartment of bacterial microbiome. So you have a microbiome in other parts of your body, but the gut is the largest compartment of that. And so this is not by accident. This has happened over millennia, that we've co-evolved with these bacteria. And there's as many, if not more, bacterial cells in your body as there are human cells. And they express more genes, produce more proteins than our own cells. We depend on them. If we didn't have the metabolites that they produce, we would not do well. They don't live in our bodies rent-free. They do stuff for us. And when things are good, it's a symbiotic relationship. They benefit us and we benefit them. We give them a safe place to live. But when things are off, we call it dysbiosis. And if you follow this field, you've probably heard that word a lot. But it's a very general term. It just means the balance of bacteria are not normal. And this can give rise to symptoms. And we've seen this potentially cause other diseases. And there are many, many studies out there that have shown that in people living with Parkinson's, that their gut bacteria is off, that they have dysbiosis. It's still unclear, though, is this causing Parkinson's? I think we make a jump sometimes and say, well, they have dysbiosis, that caused the Parkinson's. It's not clear, because it could be the other way around. It could be that your GI tract is moving slowly, and that throws off the bacterial balance. So there have been a lot of studies in mice, and some of them are really impressive and have shown some really exciting things. But overall, we can't make too many conclusions in how that translates to humans. And ultimately, we're at the stage now of the research where we're doing studies in humans. And I think for this talk, we'll focus on that. And so one of the things that people do with regards to the microbiome of Parkinson's is, can we replace the bacteria? If you have dysbiosis, let's just get rid of the bacteria having and replace them with a better balance of bacteria. And we can do this with something called fecal microbial transplant, FMT. You're going to hear FMT a lot in this talk. And the basic idea, I know we're all eating breakfast, but the basic idea is that you get a sample of stool from a normal, healthy person, and then you transplant it into a person who needs it, who has some condition that you're trying to treat. The only approved treatment for FMT right now by FDA is for an infection called C. diff. You might have heard of it before. And then the thought is that once you transplant this normal, healthy microbiome, it takes over and your disease goes away. So, I mentioned right now it's only approved for C. diff, and we've known this for a long time. This is not new. But there's a lot of research in this field applying it to other diseases like inflammatory bowel disease, ulcerative colitis, Crohn's. But then you can see studies where they apply it to everything, from asthma to multiple sclerosis, et cetera, et cetera. There's a lot of enthusiasm for FMT, but we're still catching up to see if it works. Now, regarding Parkinson's disease, there are different levels of evidence, and in the past there were a lot of uncontrolled, non-randomized studies, but more recently, there have been a few randomized controlled trials that have come out, and I'm going to focus on those that have come out in the last year or two. But even with these better quality studies, there's still small numbers of patients. We're talking like a dozen or 2 dozen patients in each study. And so this one came out in 2023, and it came out of the US. And they tested FMT in Parkinson's. I'll just break down the results real quick. So there was the total number of patients was 12, not a lot of patients. 8 of them got FMT, the rest got placebo. But they weren't really well controlled in terms of making sure that they were similar in terms of the patients who got FMT and not. The people who didn't get FMT actually had worse cases of Parkinson's. And so FMT, I didn't mention, can be given multiple ways. It can be given by capsules, through an NG tube, through an enema, through a colonoscopy. So that's one of the issues that makes this difficult to study and compare to other studies. So you'll see that in this study, the FMT was given as oral capsules over several days. In general, FMT was safe. Nobody got any serious infections or serious side effects. And they did show that the FMT improved the dysbiosis, but it did not objectively improve the motor symptoms. However, patients subjectively felt that motor symptoms were better, and that's worth something. But this is not a silver bullet. And on some measures, they had improved GI function. Again, this is a very small study. More recently, this study came out of Belgium, I believe, in the last year, just a few months ago. Another larger controlled trial came out for FMT and Parkinson's. And this study was 22 patients got FMT, 24 got the control. And in this case, the FMT was administered through a nasogastric tube, meaning the tube was placed through the nose down into the stomach. And they got 1 dose of FMT. And again, it was safe, mild side effects at worst. And the top line result was that at 12 months, there was a statistically significant improvement in motor symptoms. So this is something to be happy about, but it wasn't a huge difference. So when they measure these, in these studies, they'll have a movement disorder specialist do a lot of exams and then score them. And the improvement was 3 points. And so that's better than the controls, but it's not a huge difference. This didn't cure Parkinson's, but there was some positive effect. And I should also mention the GI symptoms didn't improve in that study. It was just the motor symptoms. This is the last study I'll talk about. This one came out of Finland, and another randomized controlled study of FMT and Parkinson's. This came out a few months ago. And so in this study, 30 patients got FMT, 15 controls. And in this case, they gave it via colonoscopy. So you had to do a bowel cleanse and then do the colonoscopy, and then they administered the FMT that way. And they selected patients who only had dysbiosis to begin with. They tested them first because they felt that if we prove that they have dysbiosis, these are the people to treat. And then the big surprise was they didn't get better. And in fact, the placebo control group got better. That was very odd to them. They had some theories that maybe just going through the bowel cleanse before the colonoscopy might have been what was the benefit. But this was, a very, surprising and disappointing result. But it just goes to show that we're in very early days of FMT and Parkinson's. And, I wouldn't jump the gun and say this is going to be, the cure tomorrow. I think there's still a lot of things we need to sort out. Which type of donors, so this is the headline, exactly. I see cases like this in clinic too, by the way. And like what type of donors, how are you going to give capsules at G2, colonoscopy, do you give antibiotics before, how long do you give it for, do you redose? We're still sorting all these things out. And so, this is a kind of a funny example where a patient on his own did FMT from his mother and then got her menopausal symptoms, supposedly. And so I've had patients who have done FMT at home, and I can tell you none of them had their Parkinson's cured from that, and they had other issues as a result. So don't do that, is my advice. So we talked about replacing the microbiome. So some studies have looked at removing bacteria with antibiotics, for example. So H. pylori is a very common bacteria that can cause gastritis in your stomach, and it can cause ulcers, and it can even cause gastric cancer. So this is an important bacteria to pay attention to. But this has been studied a lot in Parkinson's, and there's been mixed results. Ultimately, I don't recommend, thinking about H. pylori as the way to treat Parkinson's. But if you have GI symptoms, you should be tested just like anybody else for H. pylori. You should be treated for it because it can prevent you from developing complications like ulcers or even cancer. So something to pay attention to. Then there's something called SIBO, or small intestinal bacterial overgrowth. This is not an infection per se, but that you have too much of the bacteria in your small intestine that's normally there, but there's just too much of it. And this is likely due to dysmotility, because you're not doing your housekeeping and sweeping those bacteria out as often. And so I'm not usually a big fan of just treating everyone for SIBO. Usually I try to treat the underlying dysmotility. We can talk about that later, but if you treat the SIBO and don't treat the dysmotility, the SIBO just comes back. And whether or not this improves the Parkinson's symptoms, there are some small studies that show that it may improve Parkinson's symptoms. But it's not really clear if this is directly because of the SIBO or because you're improving the GI issues. I talked about this already. And so we talked about replacing, removing, how about adding bacteria, adding the good bacteria, the probiotics. So there are many, many studies, and you can cherry pick the ones that suit your, baseline views. But, most of them are not that great of quality studies. And then when they do things called meta-analyses, where they pool all the results and try to get the average result that comes out, it's kind of a wash. It's just insufficient evidence to recommend it. And for me, one of the main issues is that we don't know exactly what's in these probiotics because they're not normally FDA regulated. They're treated as a supplement. And so that makes it difficult to know what you're actually getting, if anything at all. And we don't know how much you're getting. And we don't know which strains are the best for Parkinson's. So I don't normally forbid a patient from taking probiotics, but I tell them, it's probably a waste of money. It's probably not going to hurt you, but it's probably not going to help you either. And probably if you're really motivated to work probiotics, think about getting probiotics naturally from your diet. So these are some examples of foods with probiotics naturally in them. So then there's this idea of prebiotics, and so you can feed the good bacteria that you already have. And so there's, very early evidence that maybe this is helpful. And I would, for me, people ask me about this, I would say, yes, of course, prebiotics. This is mostly fruits and veggies and fiber, things that are healthy for everybody to eat, that we should all be eating anyway. And so if you're motivated to try to do things dietary-wise for your Parkinson's, this is a great way to go about it. So just some takeaways. It may or may not play a role in the microbiome in Parkinson's, but this has really captured our imaginations. And I think because, first of all, we feel like we can wrap our heads around it. It makes sense to a lot of people. But also, I think there's a lot of desire for new solutions in Parkinson's, because it's been a long time since we've had some really good new treatments. And then there's different ways of trying to manipulate the microbiome, either replacing it, removing it, supplementing it or feeding it, essentially. But none of these are fully proven yet. I think FMT is making some headways, but still that has a long way to go. And the real takeaway I want is that if you're pinning your hopes and dreams on the microbiome being your solution for your GI symptoms, don't wait for these things to develop, because it might take years. There's things we can do now to help your GI symptoms and potentially help Parkinson's in general. And so, I would say, don't discount the things you can do today with GI issues. And then I always make this plug, make sure your colon cancer screening is up to date. People always forget about that. And so you don't want to miss something like a colon cancer that can be totally preventable. It's one of the leading causes of cancer in this country. So that's my email address. I'm happy to chat via email, and also happy to take questions. I have other slides that go over more clinical aspects like symptoms related to Parkinson's in the GI tract, but I think it might be better if we go by questions and then go from there. Thank you. Okay, so let me jump ahead.
What's good for the dysmotility?
Yeah, so I'm going to do a little bit of a talk here, a long-winded answer. So dysmotility is a broad range of symptoms, a broad term to cover a lot of symptoms. And so these are different symptoms, and these are different organs that can be associated with those symptoms. And you can see there's some overlap. I'm going to just get through this. But there are many medications that we use initially. First of all, you can start off with over-the-counter stuff. And it turns out a lot of these medications work well. And just because they're over-the-counter doesn't mean they're less good. And I often start patients in my clinic on things like Senna, which is, you can get it in a pill form. Some people like it more in a tea form. I prefer the pill because you know how many milligrams you're getting. It's more consistent, and I think it's probably more convenient. And this is something that will speed up your colon motility. Things like this or bisacodyl. Now, people also take a lot of magnesium-based drugs, like melted magnesia, for example. I think that's okay if you're taking it, once a week or less. But the one thing I get concerned about is if you're taking it multiple times a day, every day, and it's not being monitored by your primary care doctor, that you can have a buildup of magnesium in your blood. And that can be dangerous, especially if you suddenly develop a kidney issue. So it's not my favorite because there's better options. Now, in terms of, if those medications don't work, the one I usually turn to for motility is something called prucalopride. The brand name is Motegrity. And so this is a great medication for Parkinson's patients with dysmotility because although it's technically approved for constipation, it actually speeds up motility from top to bottom. So even if you have small bowel, dysmotility, if you have gastroparesis, it can help. So this is a really great medication if you have those issues. But you might not require this medication, but it's a good one to have in your backup.
I have a question for you. I personally, I'm a physician, and I personally promote diet as a disease-modifying lifestyle change, if it needs to change. And so, of course, modifying that microbiome with diet as opposed with probiotics or fecal cell transplant is fecal transplant, not fecal cell, but it's obviously something that we're waiting for better results. And you did mention how to promote good bacteria. What about starving bad bacteria? What is your take and what I read about animal protein and even dairies in terms of reducing that in order to starve the bad bacteria, not just help the good bacteria taking their place?
So that concept of starving the bad bacteria is not one I'm very familiar with, to be honest with you, but it's an interesting concept. But I would say in terms of dietary issues, like dietary interventions, some of the best data that I'm aware of is with like Mediterranean-style diets that have been studied pretty well in Parkinson's. But also, I always like to remind patients too that this idea of nutrition and dietary interventions is like a whole other field, and there are specialists even just in this. And I often work closely with GI dietitians because you can spend a whole hour visit just talking about the diet, and so it's a very in its own right
that's a major need. Most dietitians these days take care of losing weight or diabetes in general, and we don't have Parkinson dietitians. Most people come back to me and say, the guy just wanted me to lose weight, which is not necessarily what we want, especially with Parkinson.
Exactly. Yeah. And so to that point, so I always give a heads up to the patients. Like, not all dietitians are the same. Like, there's general dietitians for diabetes, weight loss, kidney issues, heart failure. But like what patients probably really need is a GI dietitian. And there are some places where they have a really well-developed GI dietitian. The people I work with most closely are at UCLA. And there are people there that are well-versed in Parkinson's just because of their own personal experiences, not something they learned from a textbook. And I work with them the most closely.
Can you talk a little bit about how these problems with motility can impact our medication uptake?
Yeah. So this is related to something I see a lot. And specifically, I'll take a step back. So with Parkinson's medications, specifically the carbidopa-levodopa-based medications, we always advise patients to take their medications away from food by about an hour or so, depending on who you talk to. And the idea with this is that the protein in your diet can interfere with the absorption of that specific medication. Now, when we tell patients to, take your medications like an hour before or an hour after a meal, we're presuming that your intestine is moving normally and your stomach is moving normally, but that's not often the case, and that's what I see a lot. And that specifically, if your stomach has delayed emptying or gastroparesis, that this is a big problem. And so people might, have not eaten for an hour or two, but there's still actually food in their stomach, and we see that a lot. And so this can lead to issues with erratic absorption of drugs, and either they don't kick in, or they're not effective enough, or there's dyskinesia suddenly because they're suddenly absorbing too much if they get too high of a dose, because the presumption was it wasn't enough. So I get a lot of referrals sometimes from patients who don't have overt GI symptoms. They're not complaining necessarily of GI issues, but then when we talk, and they reveal like, I can't eat a full plate of food anymore because I just feel really full. And there's these other sorts of issues like early satiety, things like that. When we do some tests, we find that their stomach is actually moving more slowly. And then when we start a pro-motility drug, like prucalopride, for example, then suddenly they're absorbing their drugs too much. And that's almost a good side effect, because that means they can go down on their medications. But I have to warn them. I'm like, hey, if you suddenly get dyskinetic after we start this, that means your stomach was moving too slowly and we fixed it. Now you're going to suddenly absorb your medication better. So this is something we see all the time.
I read something recently about the relationship between biomes that produce vitamin B2 and B7, and the interaction with Parkinson's disease. And I was just wondering if you could comment on the B2 or B7 deficiency and how it might relate.
Yeah, so I'll be honest, I'm not too familiar with that association between B2 and B7. Definitely there's a relationship with B12. And so that's something I'll look into because that's interesting to learn. But I'll make a plug about B12, is that oftentimes I'll get patients who are diagnosed with B12 deficiency because their PCP is just checking the labs, the routine labs, but then they don't know why they have B12 deficiency. Like if you're like a very diligent vegan, you could perhaps get B12 deficiency from dietary issues, but most people aren't. And if you have B12 deficiency, I often like to find out why. And so there's non-Parkinson's related reasons why, and we can talk about that later. But also specific to Parkinson's, there could be issues with SIBO, for example. If you have bacterial overgrowth in your small intestine, they can interfere with your B12 metabolism. They can basically steal the B12 before you can absorb it. And so those are things that I would encourage people to pursue, is like find out why you have B12 deficiency. It could also be related to Parkinson's drugs too.
In your studies that you talked about earlier, not your studies, but the studies that were mentioned, that help Parkinson's symptoms to some degree, not a lot. Did it show any type of slowing progression?
In that second study, they did show less of a, I guess you could call it a progression. Like the scoring of the motor symptoms was better if they got the FMT, but just slightly. And so that degree of change though was so small that it's not clear if a patient would notice that. They may or may not. It might be so slight that maybe a clinician on an exam might notice it, but it's not going to be necessarily a life-altering change. And so I think we're, again, like, you know, I always say, like, I want to take a step back and say, you know, for me, I'm not, I try to, I know I always say curb your enthusiasm about the microbiome and Parkinson's, but at the same time, I don't want people to think I'm anti-microbiome, because I think I'm just trying to be a healthy skeptic. And I think we'll get there with time. But it's just, we're working those details out. Yeah.
We have time for one more question.
Hello, Mike and I came to see you recently, and one thing that you said that was very interesting to me, and I think would be interesting to a lot of people, is that by the time you feel constipated, you've already been constipated for a long time, probably already before you notice it.
Yeah.
So, like, based on what you had talked to us about, we've taken a much more aggressive methodology for avoiding constipation. Mike's had it for 25 years, but this is the first time we're really focusing on clearing out and being sure, using the Miralax and all of that. And I just think that most people probably that aren't aware that by the time you feel it, you're already probably super plugged.
Yeah. So yeah, I don't want to take too much time to get to the slide, but essentially there have been studies on this, and they've measured like how much constipation does someone need to feel it symptomatically. And so you have someone without Parkinson's, and they'll feel constipated after their colon's a little bit distended. But then you have someone with Parkinson's, and then that same amount of distension, no symptoms. And then by the time they do feel symptoms, it's really severe. And so this can cause problems down the line. And so that's why I try to be more proactive. And I think, you know, even if someone's not necessarily feeling that symptomatic, I've noticed treating that, the patients come back and say, you know, I just feel better in general, you know, or things are just, I feel better in general. So I think it's worth treating that.
Thank you so much, Dr. El-Nachef. Thank you.
you.
So we're going to bring up Dr. Xenos Mason, the role of genetic testing in Parkinson's disease care. Give a warm PCLA welcome to Dr. Xenos.
Good morning. So there's a lot of familiar faces here. So for those of you I haven't met, my name is Xenos Mason. I'm a movement disorder specialist, and I've been involved with PCLA for a few years. And it's always an honor to come and speak to you and be involved. So thank you to all the organizers for having me again. Genetics in Parkinson's disease. So I'm not a geneticist, and this is early days for a lot of these concepts. As genetics becomes a little bit more integrated into our practice, we're learning how to integrate that information and how to understand what the genes are doing. So I want to introduce you to some of these ideas today, and then have a little bit of a conversation about how we can think about genetics in the care of Parkinson's disease at various stages in various ways. So important disclosures. So we're going to talk about the 3 ongoing initiatives right now: PD generation, the PPMI initiatives, and then the GP2 initiatives. Talk about the types of genetic testing that you might undergo or that can be done, and then the implications of genetic testing for first screening and family planning, diagnosis, and then prognosis, which is predicting how the disease might change over time or what symptoms might happen. And then I'm going to leave implications for treatment to a later talk with Dr. Tagliati, who is much more well-versed in at least a couple of the genes that are being targeted right now in clinical trials. So we're going to talk to you, or he's going to talk to you about that towards the end of the day. So PD generation is one of the 3 initiatives. This is from the Parkinson Foundation and also the Parkinson Study Group, which is a group of academics who are studying Parkinson's. This is an initiative that so far has sequenced about 8,000 people with Parkinson's disease. And they're focusing on a subset of genes. Last year, they just published their first results from the genetic testing program that they're doing. So of these 8,000 people that they have tested, They found mutations in a certain number of them, and they've published papers on what those mutations are. Those are mutations that they were specifically looking for. But PD generation and a lot of the studies that have been done so far are great, and 8,000 people is a good number of people to look at in terms of their genetics. But a major issue has been selection of people. So in clinical trials in general in Parkinson's disease, they're very enriched for certain populations. You see here in dark are the people that they've sequenced, and you see that it's very focused on urban areas. And that's fine for us in Los Angeles. That's also where a lot of the academic centers are. But you do end up sequencing a lot of white people, a lot of relatively wealthy people in these urban areas, and you don't get the whole genetic landscape of what the genes and the people with Parkinson's disease might look like and what their genes might be. So for example, there's Black African American populations that have different genetic risk factors for Parkinson's disease. There's been studies done on those populations that show that some of the genetic changes present in those populations are not present in the white populations that are typically sequenced in these studies. So along those lines, the Global Parkinson Genetics Program, or GP2, is another initiative that's now hoping to sequence 150,000 people world over in a much more diverse fashion. So that's going to give us a much better idea of the genetic landscape or the changes that are present in the genes across many, many more people with Parkinson's disease. And the third initiative that I want to tell you about is PPMI. So genetic testing within the Parkinson's Progression Markers Initiative has been going on for a number of years, and it's just one aspect of the study. But this study is looking at people with Parkinson's, people then with genetic forms of Parkinson's, and a whole other population of people who have risk factors for Parkinson's, specifically symptoms that often happen before the onset of PD. So that's REM sleep behavior disorder, a dream enactment, or a loss of smell. And by looking at these three populations, PPMI is understanding what genetic changes might be present in all three and comparing those changes to results in different diagnostic tests that they're running across large populations of people. So in particular, they're doing DAT scans or dopamine transporter scans in large populations with Parkinson's or prodromal Parkinson's. And they're also testing for abnormal levels of protein in the cerebrospinal fluid. So I'm going to go over each of those tests. But PPMI is really interesting because it can now, by testing the genetics and perform these diagnostic tests, compare the genetic changes to the changes in the tests and see how those things might be related. So I'm going to talk about that in a few slides. So of these three initiatives, and over the last 20 years, there's been about 15 to 20 genes that have been implicated in Parkinson's disease, depending on the strength of evidence and the amount of confidence that you have in the evidence. So SNCA was the first one. This is a synuclein gene, and this is very rare, but when it's present, very often causes Parkinson's disease. This was the first gene to be identified about 20 years ago. On the other side of the spectrum, this is a very common gene, glucocerebrosidase, GBA1, very common gene in the population, and it's present in up to 15% of all people with Parkinson's disease. But unlike the SNCA gene, which almost always causes Parkinson's disease when it's present, the GBA1 gene is a risk factor. And so if you have this gene, or two copies of this abnormal gene, you have a higher risk of Parkinson's disease, but your risk is still nowhere near 100%. So it's a different, this is two genes that illustrate two sides of the spectrum. At one side is a gene that's very likely going to give you Parkinson's disease if you have it. On the other side is GBA1. LRRK2 is another important risk factor gene, which is present in many people with Parkinson's disease, but it's also present in lots of people who don't have Parkinson's. So it increases your risk. Parkin and PINK1, I group them in the same sort of bucket. These are genes that are involved in function of the mitochondria. These are the power plants of the cells. And these gene mutations are often found in young onset Parkinson's disease. DJ1 is another mitochondrial, and then VPS35. So I just list these 7 because these are the 7 that are being tested by the PD generation program. And these are probably the 7 most important genes in Parkinson's. And it also gives you just a sense of the spectrum of genes from very causative to risk factor gene. So now you know the genes. So what if you want to get tested? So if you undergo genetic testing, you'll undergo one of three kinds of tests. One is a panel test. So panel test is the simplest. That will look at those 7 genes that are most well associated with Parkinson's disease. It'll look for abnormalities in only those 7 genes. So the advantage of this is that it's inexpensive and it's relatively easy to interpret. But you don't have to just look at the genes that we know cause Parkinson's disease. You can actually look at all of the genetic material as well. So there's a kind of testing called whole exome sequencing or whole exome testing. And this is looking at all of the DNA in your cells that forms proteins. So just to remind you, DNA is transcribed into another molecule called RNA. And RNA is used as the template to create proteins. Proteins are the workhouses and the bricks of your entire body. It's how your body does everything it does. Everything from contract the muscle of the heart to respond to light in the eye, to form the skin. Proteins are the basis of all those functions, and DNA to RNA forms proteins. But there's a lot of DNA that actually doesn't encode proteins. It's non-coding DNA. And you can test that DNA as well. And that's where we get into whole genome testing. So when you've heard of the Human Genome Project, that's because the genome is comprised of DNA that forms or encodes proteins, but also a lot of other DNA. So what does this DNA do? It changes the way that the genome is expressed. It changes the way that the DNA that does encode proteins is expressed and how often it's expressed. And so that can change the activity of coding portions of the genome. So hopefully that makes sense. This whole genome testing is the most expensive and it's the most complicated. Because if you have a genetic mutation in a part of the gene or in the part of the DNA that doesn't encode a protein, you don't always know how important that mutation is. It could be that that is meaningless, or it could be that that's very important in the DNA and the process of transforming DNA into protein. So this is where expertise as a geneticist comes in, or a genomic counselor. So now, if you get tested and you are positive for one of these genes, what do you do? So I want to talk about a few concepts that are important to how you would think about this in terms of your family planning and its relevance for you in general. The first is inheritance patterns. So that some of you might have seen this. This is a pedigree. And in these diagrams, the cross-hatching represents people. So the squares and the circles are males and females. When you have the gray crosshatch pattern, that means that there's a carrier of an abnormal gene, but that person doesn't have a disease or a disorder as a result. So when 2 carriers get together, they can have kids that share both copies of the abnormal gene, one from each parent. So this is an autosomal recessive form of inheritance. Maybe you've heard this. Blue eyes are inherited in a recessive fashion, where both parents generally need to have blue eyes in order to have a blue-eyed kid. This is just one type of inheritance. Autosomal dominant inheritance is another one. Maternal inheritance or paternal inheritance can happen. But I just don't need to get into the details, but just to illustrate that when you have a gene, you don't always know if it's going to be inherited in a recessive pattern or in a dominant pattern. And that completely changes the relevance to you and your family because it changes the risk for any kids or offspring. So even apart from that, even if you have a kid that has 2 copies of an abnormal gene, there's another important concept, which is penetrance. So here you have basically when you have affected people, see if I can use this. You have the fully black colored square. In fact, that's not really how it works because genes have what's called incomplete penetrance, where even if you have 2 copies of an abnormal gene, you don't necessarily have 100% risk of developing a disorder. So just like in the GBA1 gene I was talking about before, it's a risk factor for Parkinson's disease. Part of that is because GBA has an incomplete penetrance. So it might only cause disease in this case, 60% of people. So then if you have complete penetrance, you still have variable levels of expression of the gene or expressivity. So because of this, there are, even if the gene is 100% penetrant, different levels of disease because of expression of the gene. And when you combine penetrance and expressivity, you have a very wide variation of symptoms, even from the same abnormal gene. Okay, so this finally, the last concept is pleiotropy. One gene can cause lots of different things, not just one disease. GBA, for example, can be a risk factor for Parkinson's, and it also causes a different disorder called Gaucher's disease. So when you combine all of this, how do you take one result in a test and make any sense of it? If you have a GBA mutation because of inheritance and penetrance, expressivity, pleiotropy, all these words that are not important to remember, but just to remember the concepts that these things are complicated. How do you know the relevance for you or your family? That's when a genetic counselor comes in. That's why genetic counseling is so crucial to genetic testing in general. And it's important to always have that available as a resource. So indeed, PPMI, for example, when subjects are tested, they can get free genetic counseling as part of the testing. PD Generation also provides that service. They'll do free testing for you if you sign up, and then we'll provide you with free genetic counseling. Because they don't want to just give you a result and leave you out in the desert with no way to interpret it. So it's really important that you make use of resources and genetic counselors. Those are usually available in an academic center where you might find a movement disorder specialist, but they're also just available in the community, and you can be connected with genetic counselors through PCLA and through your movement disorder specialist. So now switch. We'll talk about how genes affect Parkinson's disease diagnosis. So most of you are likely aware that Parkinson's disease can be diagnosed without any additional testing, just based on your examination and what you tell your doctor and how you look and what's happened to you in the past, your history. But when we do use additional tests, there are two that are most important. One is called a DAT scan. So who's heard of DAT scans in the room? Just raise your hand. Okay, great. So I don't really have to explain what a DAT scan is, but there's a radioactive tracer, for those of you that haven't heard, that's injected into the blood, and that binds to areas of the brain that are important for dopamine. And when you have reduced binding, that shows up as a less bright area on the scan here. And that can tell us that there's a disease like Parkinson's disease that's present. Can't always tell us that it's Parkinson's disease itself, but in the family of Parkinson's disease. So it's not a hugely useful test, but it can be a useful confirmatory test. The other category of tests that are done that are coming into use now are tests that look for the presence of an abnormal protein, which is characteristic of Parkinson's disease. Some of you might have heard of alpha-synuclein. So the SNCA gene is important for alpha-synuclein. Alpha-synuclein accumulates in Parkinson's disease, and you can look for abnormal alpha-synuclein in various places in the body. So a skin test is one way to do that, and now the skin biopsy testing is becoming available. Another way to do that is to look at the cerebrospinal fluid. That's the fluid that bathes the spine and the brain, and you can extract some of that fluid using a lumbar puncture, and then look at the fluid to try to find the abnormal protein. So I mention these because, like I said, PPMI is running these diagnostic tests on all of their subjects, and they're also performing genetic studies on all of these subjects. So no test is perfect. Every test has an accuracy, an ability to find Parkinson's disease if it's present, for example. And that we call the sensitivity of the test, how likely it is to find Parkinson's disease if you have it. And that's rarely, if ever, 100%. But also genes do modify that. So there's some interesting initial data that was published by the PPMI folks last year to show that when LRRK2 mutations are present, these lumbar puncture tests are less accurate. And when GBA mutations are present, the lumbar puncture tests are more accurate. So this is just preliminary data, and just in one population, but some initial signal to tell us that some of these genes might change the way we think about using diagnostic tests in the clinic, or might change the way that we interpret the results of the tests. So really important to connect more genetic data over time in more people to the results of these tests in order to fully understand this. But this is how we're going to start to use this information in the future. Not so much right now in the clinic, but you'll see this a lot more in the next 5 or 10 years. What about how genes affect prognosis? There's a lot more interest in this, especially as people get genetic screening. They say, oh, I have this gene mutation. How does that change what I might expect in the future? And again, it's early days for us to say with any certainty whether one gene is going to cause one symptom or is going to, in general, result in a form of Parkinson's disease that progresses in a predictable way. Okay, so the general answer is we don't know yet, but there are some trends. So this is a nice depiction of the spectrum of Parkinson disease. You can have really young onset Parkinson disease here with just a few motor symptoms, and then not so many of those non-motor symptoms. At the other end of the spectrum, you can have somebody who has a lot of difficulty with gait, more difficulty with cognition, maybe a later onset of disease. And there is some correlation. So those mitochondrial genes, like I said, tend to be associated with these young onset forms of Parkinson disease that more often stay with just movement-related symptoms for longer. And GBA mutations, it seems, might be more associated with a higher risk of cognitive symptoms over time. And then other mutations, so the LRRK2 mutation or specific mutations of this gene might be associated with forms of Parkinson disease that are later onset with mostly movement symptoms, but actually relatively less cognitive or cognitive or psychological problems, psychiatric symptoms like depression and anxiety. So early signals that genes may be able to be used to predict how somebody will, the prognosis of somebody's Parkinson disease and how things will change for them over time. But at this point, there's not enough data to be confident. And then the holy grail is if you find a treatment, can you have a treatment, if you find a mutation, can you have a mutation-directed treatment where we use different medications for these different genetic mutations? So I'm going to let Dr. Tagliati talk about that in a little bit. But again, early days, but we're starting to investigate certain drugs in clinical trials that are using this strategy. Okay, so that's just a bit of a whirlwind overview of the concepts that are important to this. And then I'll just reiterate and summarize that it's early to use genetic information in the clinic to make practical decisions. But I think in the next decade, we're going to start to get enough information through these 3 important initiatives that we are going to start to understand what an individual genetic mutation for one person means or for their family. So stay tuned. But this is what we know so far.
Thank you very much. We have time for some questions.
I hope I asked this question pretty clearly. The genes that you listed there, the mutated genes, what caused that mutation?
So it's not always clear what causes a mutation. Usually those run in families and they occur during the process of production of the sperm and egg at some point in a prior generation. And they can also occur as somebody's developing in the womb. And so there's not a single cause, and it's not that a ray of radiation came down and zapped the DNA and caused that mutation, but usually just random events during the process of reproduction.
Yes. So at USC, are you familiar with CRISPR and the gene editing technology and how it affects people with Parkinson's as opposed to people who have the gene for Parkinson's?
Well, there's no, so I'm familiar with the basics of CRISPR, but definitely not my area of expertise, and I'm not a geneticist. There are no trials using CRISPR. I think that there have only been maybe 2 or 3 trials that have been reported so far using CRISPR in human beings, and that's been not in Parkinson's disease. So I'm not aware of any data or any pending studies that are using that specific strategy. But gene editing in general, it probably is not going to be applied to Parkinson's disease because the genes that are present throughout your life probably are have a long-term effect. And by the time you're developing symptoms or even prodromal symptoms, you might have had 20, 30, 40 years of the effect of that abnormal gene. And so changing that gene at that point might not necessarily, affect significant changes in the way the Parkinson's disease is going to be treated. But gene-targeted medications, though, are still reasonable because I think I'll let Dr. Tagliati talk about that a little bit more, but specific genetic mutations in certain kinds of proteins can be targeted more easily with small molecules like typical drugs, and that might be a reasonable strategy.
Good morning. So I'm kind of wondering about the relations between these 3 genes and other genetic mutations, right? So, for example, I have the alpha A1 antitrypsin mutation, right? So is that related to this, how does that tie into these mutations, right?
So I can tell you, I certainly don't know, right? But it's possible. And the genetic milieu, so the other genetic changes that somebody has, mutations, but just variants are an important aspect of the pleiotropy and penetrance of a gene. So a gene is never in isolation, right? It's in an environmental and a genetic landscape. And the presence of other abnormal mutations does affect the function of, say, a Parkinson's disease mutation or a Parkinson's disease variant, as does what you do for work and where you live and how you've lived your life and what kind of exercise you do. So generally, yes, but this is a hand wavy explanation. And it's complicated. I don't know the specific answer.
Hi. Generally speaking, Parkinson's is in an older population beyond childbearing years. And so are you looking to catch it early, or is it after the fact? And then you're going to change the way you treat?
Well, in some cases, sure, that somebody who's affected by Parkinson's disease might undergo genetic screening, find a genetic mutation, and then have their kids interested in being screened, right? So that's usually where it's most relevant, is kids and grandkids. It's, in the rare case, you'll have somebody with young onset Parkinson's disease that might still be able to have kids.
Thank you. Actually, this is fascinating. But my question is, you haven't come right out and said this, but do you recommend that people go and get genetic testing, regardless of where they are in their childbearing years? But should we go get a genetic test? Where do you land on that for just future value to our?
So the value to science is very high, and the value to the community is very high. So these are free programs, and I think to the value for learning is, unquestionable. You should sure go out and get tested if you want to contribute. Now, as to the value to you individually or your family, it's much less certain. Some of these genes are going to be are going to have a clear effect. Like, for example, you're not going to find an SNCA mutation, but if you happen to, then that's going to be a really clear answer for your family. But even a GBA mutation or a LRRK2 mutation, we know enough that speaking to a genetic counselor will give you some sense of your risk, and will give your kids and grandkids some sense of their risk if they want to get screened of having Parkinson's disease. Now, whether that's, your risk is 60%, period, or it's 5 to 10 times that of the general population, those are still relevant things to know. And so depending on the gene, there are variable levels of individual benefit, but there's a great benefit to the community for sure.
Hi, Dr. Mason. It's Simon here.
Simon.
Could you just go back to LRRK2 for a quick moment? I noted that you said 1 to 2%, and then there was a 15% number. Could you just tell me what those are?
I think LRRK2, so I think on the slide, it's 1 to 2% of the general population, but I think 15% in Ashkenazi Jews.
Oh, is that right?
And so much more and that's often the case that in specific, historically insular populations, historically separated in some way, that these genes have a higher proportion.
Right.
And so they're important, especially if, you have that kind of background, important to do the testing, and they change the way that you think about the genetic result as well.
So for someone with LRRK2, you're saying that 15% of offspring get it?
Oh, what I'm saying is actually, so across all people with Parkinson's disease, about 1 to 2%, say, of the people in this room with Parkinson's disease would have a LRRK2 mutation. But if this was a conference for people of Ashkenazi Jewish descent with Parkinson's disease, 15%.
Right. And is there any specific information on LRRK2 regarding all the words that you talked about?
Oh, of risk for developing Parkinson's disease if you have it, yes. I don't have the exact number in front of me, and it's not a 5 or 10 or 15% number, but it is an increase in the risk relative to the general population. And I'll look that up for you, Simon.
Okay, thank you so much, Dr. Mason.
2 questions. I think you've answered the one about whether or not our kids should get tested. It's kind of like helpful, but for the general population. But how often is it that there are multiple generations in a family that have Parkinson's?
Well, in general, about 15% of Parkinson's disease has some genetic basis. What percent of the people that we see in our specialty clinics have family histories of Parkinson's disease? I'd say it's probably about the same, maybe a little bit higher because, often somebody will say, oh yeah, my maternal uncle had Parkinson's, or maybe a more distant relative, and that'll give them a positive family history. So you might be looking at, say, a quarter of people on average have some family history in general. Now, what percent of people have Parkinson's disease in each subsequent generation reliably? That's very low. And those forms of Parkinson's disease, like, for example, SNCA, are quite rare, but they do occur.
My question is, and I'm not clear from the presentation, is the genetic testing gotten to the point where treatment would be different based upon what you might find genetically with somebody? I mean, as of this moment, would treatment take a different course if we went through testing and we found?
No, in the vast majority of cases, no. But I'll let our last speaker, I'll let Dr. Tagliati talk about a couple of the trials that are happening now targeting specific genetic changes or people with specific genetic changes. And so basically no, but I do expect, I would hope that in the next decade, we'll probably have 2 to 3 such treatments.
Thank you so much, Dr. Mason. Hang on.
Xenos, just a last comment. First of all, the amazing effort to really put together a complicated issue. But I think that what genetic testing will bring, in addition to some personalized treatment, is a better definition of what Parkinson's disease is. And I have a couple of slides in my presentation. We're really moving forward and beyond the tremor, the rigidity, and the bradykinesia. And as you will see, the new classifications and definition of Parkinson's disease include genetic profiling. And I know profiling sounds really bad in this day and age, but it's going to be very important to know what is your genetic background in order to personalize our approach to your treatment and to research in general. I think this is, at the end of the day, is the biggest contribution that genetic research is bringing to our field.
Thank you so much. Thank you, Dr. Mason. And this is Dr. Tagliati, who's going to be speaking with us shortly. So we're going to go right to a film, Mediflix.
Hello, PCLA community. Coming to you from Nashville, Tennessee, from the Parkinson Study Group. Sorry I couldn't be there. And just wanted to wish you all a quick holiday message from me. Hopefully you're enjoying connecting with each other. And I think, if nothing else, I've realized that connection and community is a huge part of the medicine. It has been for me. You've had some great speakers today, some of whom I've trained and are near and dear to my heart. Hopefully we can keep the message of connection and community as something that's central and something that we are intentional about preserving as we look into 2025. Hopefully we can spread connection to even folks that are even more isolated and lonely, perhaps, than within our Parkinson's community. I hope you enjoy the film. Some of the folks featured here are part of the Los Angeles community, and I'm excited to be part of that community. So wishing you the very best for 2025, and hope you enjoy the film.
Parkinson's disease is very different in every patient. If you've met one person living with Parkinson's, you've met one person.
I'm Farrell Levy. I'm 73 years old, and I was diagnosed with Parkinson's in 2016.
I'm Trent. I'm 68 years old. I've had Parkinson's officially for 14 years.
My name is Deborah Jo Doxey, and I'm 68 years old, and I've had Parkinson's for 7, almost 8 years.
I'm Kathy.
I'm Trisha.
and I've had Parkinson's for almost a year now.
An off period is when a person living with Parkinson's feel like the medicines are not working well.
For me, an off period is when things just don't seem to work. I'm forgetting things. I feel very tired, and I'm so used to moving and doing the things that I need to get done that I get frustrated at the fact that I'm just not myself.
An off period is when I'm aware and know I have the disease. When I'm on, I can forget I have it.
For me, an off period is when I start to become rigid again. My movements become more limited. My walking becomes more shuffly. My arms don't move well. I start to be aware of not feeling like I have a spring in my step.
I do feel like I'm two different people when I'm comparing on and off, and I see it when I walk down the street when I'm on or off, and I see it in terms of how people look at me. I know when I'm going with, that full stride, no symptoms, confidently, and no one's looking. If I'm going on an off period where I'm walking a little more slowly, the gait is a little bit shorter, particularly if I go out in the morning first with the walking stick to try to get my body moving. There's kind of that look. They're trying to figure out what's going on with me, like what's wrong with him, what's he dealing with. That's when it's really different, and that's when it's like, I don't want to be that person.
Generally, the symptoms that people manifest motor-wise with Parkinson's can include things like tremor, stiffness, and slowness. And so those main cardinal symptoms can be the things that reemerge when people say that they're off or that their medications are not working well. Other people may describe things like, I feel like I'm in a fog. I feel like I'm having mental slowing, that their heart rate's increasing, that they're having trouble breathing even sometimes, this sort of anxiety kind of manifestation.
Oh my gosh. In the very beginning, you don't know what's happening to you. I remember at one point sitting down, looking around and just feeling a bit lost and alone, and just didn't understand it, and that was scary.
An off period does affect me mentally. It's like, okay, I'm just gonna slither into this little cocoon here till I get better, and I just kind of mellow down and wait until it's over because I gotta ride it out.
This idea of diminished capacity unlikely scares me. There's this unknown out there that's a little frightening. No, sorry, it's more than a little frightening. When it gets tough. I haven't had to answer that question quite in that way before.
It's okay.
My mom has always taken good care of me and my family. And she's always taken such good care of herself. And it's hard for me because I know that she very much likes her independence, and she's always been the person to step in and take care of others. And I think it's really hard for her sometimes to be taken care of. Mm-hmm. And she worries about that. Yeah,
We'll get to it.
We'll get through it.
Yeah, we will. Not a problem.
Mm-hmm.
There's a lot of hope and a lot of ways that we can treat Parkinson's to really help people to live, better lives and really to thrive and flourish.
My doctor has been extremely helpful the whole way.
My movement disorder specialist, that's a critical relationship for me, one of the most important in my life.
It is just so important to talk to my doctor because I'm the only one that can give him the clues as to what's actually happening.
It's important that family members participate in those discussions because we get to witness what their life is like. We're all working together as family, as patient, and as doctor to make sure that, her quality of life is as good as possible.
Working with your physician to find the right sort of bespoke combination of therapy for you, which not only includes pills, but includes sort of the bigger picture of having that person help you to feel like you are part of the treatment paradigm, not just passively receiving things, is actually a huge part of the medicine.
When my off period's under control, I feel like I don't have Parkinson's. I feel like I'm what I was before.
When I'm on, I challenge you to tell me that I have Parkinson's, what my affected state is, and then I'm 68 years old.
I just got back from a vacation in Patagonia where we hiked 10 miles. I felt great. You feel empowered when you do something like that.
I like being able to go to the house and help you out. I like to be able to talk to the girls and enjoy them. So that's my greatest motivation.
Why limit yourself? Why limit yourself to black and white?
I got a lot of living to do. Yep. And a lot of buckets to kick.
Make sure that you talk to your doctor.
Stay on. I really feel that.
Spend more time on than off.
I want to be on.
Love you, Mija.
I love you.
Wasn't that moving and wonderful? And we're so fortunate, Patricia Chavez. So we got a movement segment here with Emily Ramon and Mackenzie McCoy from Reactive Physical Therapy and Wellness. Give them a warm welcome. They're awesome.
Hello. My name is Emily. Thank you for the introduction. We're from Reactive Therapy and Wellness. So if you don't know us, we have a big team of physical therapy, occupational therapy, neuropsychology, speech therapy as of Friday. So that's exciting. And wellness, which includes our group exercise classes, personal training, and yoga therapy. So we've got a lot going on, but today we are here for a movement break. We have a clinic in West LA and we have a clinic in Torrance, but we also have a huge online program and service. And we have a table out there if you have more questions. You guys want chairs? Yeah, we grab one. You can, movement break can be seated or standing. We're going to show kind of both options. This is Mackenzie. She's a doctoral student from USC, and she will get us kicked off.
Hello, everyone. Nice to see you all. We're going to start with our warm-up song. So if you all want to stand up, find some space in the room. You can do everything seated as well if that's more comfortable for you. We'll be moving around a little bit, so spread yourselves out. And for our first song, Emily and I are just going to be leading a warm-up, so go ahead and follow along with us, and you can go ahead and start the first song. All right, let's start.
for Parkinson's. They're amazing. They're back there. So, don't hold me. It's going to be a mix of power moves and boxing. Again, seated or standing options, but I just wanted to do a quick review. Some of you may know these, some of you may not. Things you'll see in the next song. You might see a power up, which is going to almost be like you're sitting down in a seat, and then you're opening up nice and wide. Okay, like you're sitting down in a seat, reaching forward, opening nice and wide. Mackenzie will show seated version. All right, so that end game of that nice tall posture. Beautiful. We're also going to have our power rock, which is going to be like shifting your weight to one leg, reaching up towards a corner. Come back to the middle, shift the other way. Beautiful. You can choose to float that back leg, or you don't have to. You can always use support on a chair. Nice. We have our power twist. So you're going to open up both arms really wide, and you're going to twist. So your hands meet one side, open up to the middle, other way. Beautiful. There you go. And our last one is going to be our power step. We'll do it in multiple directions. So it's a big step as if you're stepping over an obstacle, and you're going to bring your hands out wide when you do. So if we go forwards, like a big step, and then we can go other side. Good. Same thing would happen sideways. Nice. Other side. And then backwards. Good. Awesome. You all are pros. Okay, we are going to throw some boxing in. Simple punches. You're going to have your straight punches. So just your 1s and 2s. Good. You're going to have your hooks. So your 3 and your 4. Good. And your uppercuts. 5, 6. Cool. I'm going to announce them as they come, but we're going to kind of flow along to the rhythm of the music. So if y'all are ready, hit it. Next song. So this first part, just freestyle. Let the rhythm move you here. Just share. It gets me hyped. All right, we'll start down. Up. Good. Yes. Down. Up. 2 more. Down. Up. Down. Up. Let's rock. Side. Side. Yes. Good. We're gonna twist. Twist. Open. Good. Twist. One more round. You got it. All right, meet me back on 2, 1, 2, 1, 2. Good. 1, 2, 1, 2, 1, 2, 1, 2. Keep going, I know. Now we got it. Hook, 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 hook again. 1, 2, 1, 2. Hook, 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 hook. Uppercut. Hook, 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 last. And hook, 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 round of applause. Okay,
Great work, everyone. We're going to do a short cool down and then we'll be done for today. So go ahead and play the next song. Alrighty, open up your feet about hips width apart. Go ahead and drop your head down to one side. Open wings and learn to fly. All in, drop down to the other. You were only waiting for this moment to arrive. Back to center, one corner of the room. Blackbird singing in the meadow. Other side. Take these sunken eyes and learn to see. Come back to center, all your life. You were only waiting for this moment. Slowly lower your head down. Blackbird fly. Come back to center. Go ahead and start with some small shoulder rolls back. Into the line of the dark black night. Place your fingertips on top of your shoulders. Medium circle rolls. Straighten out your arm. Big rolls back. Blackbird fly. And let's reverse. Big shoulder roll forward. Into the line of the dark black night. Fingertips on top of your shoulder. Forward roll. And lower your arm down. Small shoulder roll. Great work. Everyone go ahead and find a seat in their chair. We'll do one seat and stretch to finish us off today. So sitting down, Emily can show you. Straighten one leg out in front of you. You were only waiting for go ahead and lean forward towards your toe. You were only waiting for this moment to arrive. You were only waiting. Come up and switch legs. Take your other leg out in front of you. Hinge forward at your hips. Come back to center. Give yourself a round of applause. Nice work, everyone.
All right. Thank you so much. So we have Dr. Melita Petrossian, who's going to be joining us remotely, speaking to Parkinson's, dementia, and delusions, the impact on the relationship of communication between PWP and care partners. What a great topic. Thank you for joining us, Dr. Petrossian.
Thank you for having me. I really am so excited to see virtually and in person everybody dancing and moving. Today's going to be a little bit of a harder topic, and I always like to start by reminding people that not everybody will experience dementia or delusions, and it can be difficult to think about and worry about, but for people whose family members or friends or loved ones are experiencing dementia and delusions, is an important topic. So please bear that in mind. So we'll talk about some definitions, psychiatric symptoms, communication techniques, and psychological symptoms. So I want to start by clarifying what we use as far as terminology. Patients can often begin their journey of cognitive issues with what's called mild cognitive impairment, where there are some changes happening that go beyond normal age-related changes and cause some mild issues, but no actual disability of activities of daily living. And that's called mild cognitive impairment. As the disease progresses, unfortunately, some patients develop dementia. Now, that's a hard word to hear, and some neuropsychologists prefer the term major neurocognitive disorder. It's a little bit of a mouthful, and I prefer to clarify that when we talk about dementia, that's still also on a spectrum from mild to moderate to severe. So it's important to define in your mind what we use the term dementia for, because I believe that a lot of lay people view dementia as a very end stage kind of situation where people are very disconnected or unable to connect at all to their world around them. And that's not the case in earlier stages of dementia. So dementia just means cognitive impairment to the extent of inability to perform one or more activities of daily living. So if somebody used to be able to balance the checkbook and now they're making mistakes and to the point that they're really not able to do that anymore, that counts as dementia. They may still have good functioning in other settings. They may still be very conversational, very much capable of doing other things, but they may already be in an early stage of dementia. The term dementia is not the same as Alzheimer's disease. People with Parkinson's disease can get dementia and they may not have Alzheimer's at all. So it's important to bear that in mind. I've personally seen a difference qualitatively of the memory issues in Parkinson's being more short-term memory and less long-term memory compared to Alzheimer's, where I see more of like an eraser being taken across their autobiography. So people tend to stay more aware of who people are around them and that kind of thing, even in later stages compared to Alzheimer's disease. But there are some overlaps. Just to clarify, the term Alzheimer's disease refers to a different pathology that causes memory decline and dementia. So that's why I mean it's not the same as Alzheimer's disease. People have heard the term dementia with Lewy bodies, and I like to clarify that that is now felt to be a subtype of Parkinson's disease. And we may define somebody as having that subtype when the memory or cognitive symptoms began before or around the same time as motor symptoms, like movement symptoms. There is some good news to start off, which is that the rate of dementia and the median time to onset of dementia is probably lower than was previously reported. So this was published just this year. Typically, about 80% of people were felt to develop dementia. And I think people imagine that as being relatively rapid from the time of onset of Parkinson's. But it seems more recently from Michael J. Fox's Parkinson's progression marker initiative and a different cohort that the rate is actually lower. That at year 10, the risk of dementia was only 9 to 15%. And another study in that same study, the Penn cohort showed 27% at 10 years, 50% at 15 years, and 74% at 20 years. So a much longer time frame before the development of dementia. And again, that included mild forms of dementia. So one definition was cognitive deficits interfering with, but not precluding the patient's ability to carry out normal activities and social interactions. So I dare say some lay people wouldn't necessarily consider that dementia per se. So again, really important to bear that in mind. Another sort of imagining a good piece of news might be that, well, sometimes it's not dementia. Sometimes there are cognitive changes that look really scary, really concerning for dementia in a Parkinson's patient, but actually we go back and we look at the medication list and they've been started on a medication for their bladder like oxybutynin or a dystonia medication like Artane, which is trihexyphenidyl. These medications are very commonly causing cognitive symptoms, especially in people with Parkinson's, and they really should be avoided. Benadryl can cause an issue, but not always. Sedating medications can sometimes be a problem, and sometimes even the Parkinson's medications themselves at higher doses can cause cognitive issues. And sometimes not even at high doses, but again, it really depends on the patient. If somebody has been stable on their Parkinson's meds for many years and they're developing cognitive issues slowly, I wouldn't attribute that to the medications. But if there's been a recent change in the dosage or an addition of a Parkinson's medication and a relatively quick onset of a cognitive decline, then it's very important to discuss with the neurologist about that. It's very important to bear in mind that the term delirium is different from dementia. It refers to a short-term waxing and waning cognitive state where patients are often either ill or they have a metabolic disturbance or they have an infection or they've fallen and landed on the floor for 6 hours, they're in the ICU. These things are very common to cause delirium. Unfortunately, delirium is more common in patients who have underlying dementia, but in and of itself, it is not the same as dementia. And patients can recover quite well, sometimes with a delayed recovery, a few days, maybe even a few weeks. But as they recover, it's very important to beware of that diagnosis. Patients with severe depression, apathy, sleep issues, vitamin disturbances, other metabolic issues, and sometimes even strokes may look like they have cognitive decline like dementia, but it may not be exactly dementia. It's unfortunately also the truth that patients who do have an underlying dementia process can have their symptoms somewhat unmasked by a medical disturbance. So it may sort of be a sign of things to come in the future, even if patients recover, but not always. So bear that in mind. Cognitive domains that can be affected in people with Parkinson's-related dementia include memory, especially short-term memory, meaning that patients can often, when I ask them about memory, they say, oh, my memory's fine. I can remember, who played first base for, the Yankees in 1992. And that's not the same as short-term memory. If they're having difficulty remembering conversations or where they're supposed to be, that's a short-term memory issue. Processing speed is a very commonly affected cognitive domain, meaning that patients will be able to recall whatever is being asked, like the word or the answer to the question, but it just takes them longer to grasp that. And often that can be interrupted. So if a different cognitive thought comes in or somebody says something, that it can be very hard to hang on to that train of thought. Executive functioning very commonly affected in people with Parkinson's-related dementia. Executive functioning refers to planning, coordinating, for example, decision-making. For example, if you're having a dinner party, there's probably 100 steps involved, and you have this idea in your mind of how everything's supposed to happen, and that can be much harder in people with dementia. Language is sometimes affected, meaning finding words or using the wrong word, using simpler words than previously, or just getting confused when people are saying something. Visuospatial processing, meaning where things are in space, how to reach for things, what's around, especially while driving. This can be a serious issue. And then sometimes judgment, like, oh yeah, it makes sense for me to go up on this ladder right now. Well, no, you fell last week. Don't you remember? That kind of thing. So care partners, some initial steps to be involved, to consider is first and foremost, get involved. I know I'm sort of preaching to the choir here, but it's very important for care partners to attend appointments or call in via phone when there are cognitive issues happening, even in earlier stages of cognitive issues. Frankly, I don't mind. I actually really prefer it when people come with their care partners at every visit, even when they're doing really well cognitively, because there's actually evidence that people with completely normal memories forget about half of what's said in doctor's appointments. So it's always good to have an extra set of ears. But it's also good to start getting organized, with your person with Parkinson's doctor's visits, including neurologists, but also other ones as well. Preparing questions in advance, reviewing the after visit summary, going over the instructions together, making sure that all of the steps are taken, that, for example, call physical therapy, take care of this appointment, or this one we're waiting for a phone call, that kind of thing. It is a very common first step of what needs to happen for the care partner. It's very helpful to take notes. If you find that you can't write things down so quickly and you want to take a recording, just ask permission. Most providers are open to that. Maybe not in an early stage, but certainly in later stages, driving and financial fraud or errors are very common. So it's very important to take safety considerations. We're not going to talk too much in detail about driving. That's a whole other talk, but important to really start thinking about that and, engaging the person's clinicians to help as needed. Again, the other reason to really emphasize the importance of attending doctor's visits is to make sure that somebody other than the person with Parkinson's is aware of what's going on with the medications, know what they look like, know what they're supposed to be taking, how frequently, who's the ordering provider, the purpose, when are the refills due. Now, of course, people come home with a medication list. Oh, sure, it's all online. But if somebody is seeing different providers from different institutions, or they just happen to not be taking what's on the list, it's good to have your own list that you can reference in terms of, what time of day it needs to be taken. Very important to reduce the risk of medication errors. And again, also looking at, hey, do you really need to be still taking this medication? Maybe we can simplify the process. For many people with Parkinson's dementia, they have difficulty remembering to take their medication. So using a MediSet may require oversight by the care partner. As things evolve, the care partner may need to, be directly involved in medication administration. So it's good to start with a familiarity. So shifting roles, things can start with just needing reminders. Then care partners may need to start managing appointments and executive tasks, both health-wise and around the house. Then sort of managing the behind the scenes things can evolve into more direct oversight of the medications or finances, taking over for certain tasks. So in that context, it is really important to try to maintain other aspects of a relationship that are more co-equal, like remaining a partner in whatever domain is possible, enjoying music together, talking about, something you've watched or read or art together. Often it helps to have a physical activity that can be done together. If possible, if it's safe, to go for walks together, or doing one of these wonderful programs like Dancing Through Parkinson's. I know you guys all enjoyed that, and they welcome, care partners to be participating. And I think that's a really helpful thing, so that it's sort of a joint activity. It's a fun, almost like a date night kind of thing. Of course, again, as with all advice with these talks that we give, it's very individual. You have to take things all for, your own situation, but just ideas of how to try to maintain an equal partnership there. I also encourage people to engage the person in conversation on topics of interest. If there's a story that they like to tell, let them tell that story again, let them, talk about, a topic, even if it sounds repetitive to you, trying to engage that, I think, is important. Okay, so again, use as necessary. Communication tips. This is not going to be relevant for everybody. Some things are going to work for some people, not for others, but these are some ideas that can help. So when you're trying to talk to somebody, let's say, it's time for them to, find that paperwork that you need for doing the checkbook. But they're engaged with something else. Make sure that person is ready to hear what is being said before you try to talk to them, because that's a very common thing that, especially spouses do, where, we're like, we have our own agenda of what we need to get done. And we're like, okay, I'm just going to come in and say, I need this from you now, and, or don't forget to do blah, 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 and also call blah, blah, blah. That person, didn't hear the first 5 seconds of what you said because they really weren't ready to hear. Make sure you're in the room with them, make eye contact, call their attention, wait for a response. There can be, a little bit of longer time that's necessary to shift focus from a task to, talking to whatever it is that you think is important. And sometimes they're involved in something that they're going to lose track of what they were doing if you interrupt them. So if you can, try to avoid interrupting that task if it's possible, of course, if it's safe. Make sure to speak clearly and at a loud enough volume to be heard. Many people with hearing deficits lose high pitch hearing first, so that makes it hard for women to be heard. So sometimes we need to just lower our pitch if we can. I know that's hard to do, but sometimes that helps more than just speaking louder and louder. Also, wait an extra beat for a response. Often I find people just like asking and asking and asking, and then that person really gets discombobulated by, wait, I was trying to answer that first question, but you asked me it again in a different way, and so that just really taking a beat, being comfortable with silence, I think is really important here. Trying not to interrupt. Let's say the person is talking and they've lost their train of thought and they're about to get that word back. And then you say, wait, did you mean, blah, blah, blah? Were you looking for your glasses? And they're like, no, and then they've forgotten what they were literally just about to say because they got interrupted. So again, taking a beat, taking a breath, waiting before interrupting. In that vein, I'm going to skip down on my bullet points here. Sometimes the person really does want you to help jump in with the word or finishing that sentence, because they're really struggling and they know you know it and they're just waiting for you to finish their sentence. So I like people to agree on a signal that they can use to when the person with dementia wants help, so that you're not jumping in prematurely, but if you can see that, they're turning to you, usually it's natural, like they'll turn to you and they'll say your name, like, Mary, what was I saying? That kind of thing. So, it's important to have that agreement in place. Another technique is to break tasks down into simple steps or single steps and discuss one topic or task at a time. Don't jump and ping pong from, different things that need to happen that day. Just, discuss one thing, make sure it was heard, make sure it was, maybe called back, like repeat it back to you, and then move on. Some issues that come up with, decision-making paralysis are, again, getting overwhelmed by too many choices. So, what should we eat for dinner today? Oh, I don't know. Like, I can't think of anything right now. Or instead, you could say like, hey, should we have, fish or chicken today for dinner? And that just helps. So some people need more visual cues, like using calendars, checklists with photos or picture icons, like, on the door before somebody walks out the door, a picture of cell phone, wallet, keys, ID, that kind of thing. And then just like practicing the reminder to look at it and reference it and, check, that kind of thing. There's a really great digital clock that you can find, on it's not a medical thing, you could find it anywhere, where instead of just showing the time, they also will write down whether it's morning or afternoon. And that becomes important when people are getting disoriented to, 5 a.m. and 5 p.m. can look very similar to each other in terms of the amount of light, especially now. And then the day and date is always accurate there. Because sometimes when people have a paper calendar and you're marking off the date, you may not have marked off that date, and it can get disorienting. As best as possible, try to avoid arguing with somebody with dementia or asking incredulously, how do you not remember this? Sometimes I've seen patients, because the care partner knows that they're going to get tested, what's the date today? Where are we right now? Sometimes I've seen the care partner doing it to their loved one, and I say, you don't need to do that. Let us do that. You don't need to test them. Like, do you know what day it is today? Do you know where we are? Do you remember this? Like, it's really not necessary. You can sort of engage that in other ways. Also, try not to criticize the person's memory or attempts. Again, this is all very natural if you find yourself doing this. It can be really hard to maintain patience in these contexts. It can be really hard to cope with, but try your best. Maintain socialization if possible, but sometimes you might find that big events can be overwhelming. So consider limiting to a smaller group, like one other couple. And, that might be easier to get them to engage rather than withdrawing from the conversation. So there's other treatment for dementia that I wanted to just briefly mention. Of course, there are medications that we're not going to talk about. But again, this is why it's important as when we talk about communication, it's also communicating with the team and, discussing that as a possibility. But the cognitive therapy can be very helpful, or cognitive training. Cognitive therapy is a subset of what speech therapists do and sometimes also occupational therapists. It's the development of adaptive methods to address cognitive deficits like memory prompts, attentional cues, reorientation. And there are memory boot camps that can help for people with dementia. So really look for those resources in your community. Cognitive physical dual tasking is a way to do a cognitive challenge at the same time as a physical task, safely, of course. And it's been shown to improve gait and cognitive function in people with Parkinson's disease. So let's move on to psychosis. That's a hard word. Again, it's really scary to hear these terms, but psychosis just refers to a disconnect from reality. Typically, they begin with illusions rather than frank hallucinations or delusions. So we'll start with that. Patients can have a sense of presence, a feeling of somebody being, behind their shoulder, but that can spread to visual illusions and hallucinations. Illusions are a misperception of something that's there. People can see faces in patterns that don't have any visual faces. And the reason for that is that these pareidolia, as they're called, illusions, are actually very normal in our brain. We're actually designed to look and see human faces, from literally, week 2 of our existence. The first thing we've learned to fixate on is faces. So it's super common to see faces that aren't there. It's kind of like that the shortcut in the brain. So that's why people often will see faces or like human or animal images in shadows or patterns. And very commonly at the beginning, again, there's a spectrum of the severity. These can be mild and significant, but they can become more bothersome. Sometimes they can feel like somebody's at home living with them, or there's a lot of people hanging about. That can be really bothersome. Delusions, on the other hand, are fixed false beliefs. So these are, by definition, more serious because they are, fixed. They can pass, they can come and go, but at the moment, it's a strong belief. These can include delusions of reference, so believing that they're not at home, sometimes even to the point of if the care partner says, hey, you are at home, see, this is that photo of your family, this is your couch you've sat on for 20 years. They may say, no, this is a replica, this isn't it. It can be person, so they can believe that their loved one, isn't who they say they are. That's called a Capgras delusion. But often I've had people tell me that there's 2 versions of their care partner. So for example, there's the nice Anne and then there's the mean Anne. And they'll be in the car with Anne and they'll be talking about, oh, we just passed by that house that Anne used to live in, as if they're talking about Anne, but she's Anne. So, that's pretty common, unfortunately. I shouldn't say pretty common, but within the realm of delusions, that's a common one. This can become really, really scary to the care partner and even to the patient. It can be very scary, to think that they're not at home, to want to go home, and they can get agitated. So we'll talk about how to manage that. Paranoid delusions, infidelity is a big one. Theft, people are stealing from me. When delusions come with dementia, that can be a bad combination because people can sometimes misplace things. The visuospatial issue comes into play, the memory comes into play. So they've misplaced something and then they believe that people are stealing from them. So that can be really hard. And then there can just be a fear of abandonment, like, she's going to leave me, she's putting me in a home. And, sometimes there are conversations that these are sort of coming from some nugget of reality, but they can really be expanded upon. Okay, so how to cope with this? So for visual hallucinations, often just turning the lights on can help because visual hallucinations are often worse in the evening. Remove any triggers for hallucinations, like if there's a fan that looks like a young girl, sometimes just actually getting rid of the fan, that young girl went away. So that's important to bear in mind. So when it's a delusion, don't argue, especially if they, push back. You can try gently correcting. We are at home, honey, we are at home right now. Let's go get, let's go get ready for bed now. But if they argue back, no, we're not at home. Don't try to use logic. It doesn't tend to work. Respond to the emotional message and try responding non-verbally. So, I understand it must be really scary for you to think that you're not in your home, but you're safe here. I'm gonna keep you safe. We're gonna be okay. I know it's scary to feel like people are here in this house with us. But I want you to know that you're safe and I'm here for you. Again, like I said, don't argue. You don't have to agree with them. You don't have to say, oh yeah, I see them too. Don't have to engage with that. It's probably best not to do that. But just sort of framing it as recognizing their version of reality. I understand you're seeing people right now. I know that's scary. Let's come back to what we're focusing on right now. Let's go get some lunch. It's time for lunch now. Let's go do that now. Speaking calmly is really important. Like, again, trying to calmly, sometimes with touch, if they're not super, if that's appropriate, bring them down to like, let's just do some breathing techniques. If you can sort of almost just breathe for them, that can sometimes help too. That helps you too, because, when the care partner is dealing with somebody who's delusional, they're getting an emotional reaction too. So really focusing on taking care of your own emotional reaction first, and then trying to mirror that for the person with Parkinson's. If you can redirect, we sort of showed some examples of that. Further tips, again, verify the diagnosis of psychosis with the medical team. Sudden onset is often a sign of delirium, medication issues. When I get these phone calls, the very first thing I do is let's look at the medications. Let's check for UTI. Could they have a pneumonia? Could they have the flu? Very important to start there. Again, there are treatment options for psychosis and delusions. So especially when it's at the point where it's bothersome, it's really disruptive to their functioning, the delusions, really important to talk to the neurologist about treatment options, which is really great news that there's treatment for that. If you're concerned about having the conversation in front of the neurologist, where with the person in, face to face, you may want to send a my chart message for those of you at institutions that use Epic, or giving a phone call. Sometimes even let the MA know, the medical assistant know, like, I'd like to talk to Dr. Petrossian or whoever the neurologist is for a moment, and I like to do this discreetly so that patients aren't feeling like, we're talking behind their back. But that can be really important to get the whole story. Safety concerns, very important. Delusions are often associated with physical aggression, wandering from the home at night, trying to leave a moving car. I had a patient who left at night to go to his other family because he felt he had a double wife situation and he didn't think that he was at his own home. So he went to find the other, Mary or what have you. And he left in the middle of the night. So it can be really, really scary and dangerous. So, again, always discuss that with the medical team. For serious things like this, low threshold to call 911, call in your family support, call in, a person who's big enough to handle, because people can be very strong when they're physically aggressive. So, making sure that there's no access to potential weapons. And if you have somebody who's wandering, double cylinder door locks or other door security measures, and if they can wear a medical ID bracelet, that can be helpful. Sometimes the symptom of dementia includes withdrawing and disconnecting. And, I feel like, that sort of disconnect and withdrawing actually compounds the situation. So, sometimes people are coming to these lectures, hearing about dancing through Parkinson's, but, their loved one just doesn't want to show up. They don't want to participate. They don't want to do it. So that can be a big part of this. So how do you cope with that? Well, really trying to understand this overlap between what's anxiety, I don't want to go because I'm worried about who's going to be there. What's depression? What's apathy, meaning loss of motivation? What's I can't plan it myself, but I'm happy to go if you plan it for me. That's executive function. What's physical fatigue or medications being off, that kind of thing. And what's sleepiness? They're getting too many medications, they're too sleepy, they're not engaging. So this can be kind of hard to disentangle. They really do overlap. Good things to talk about with the neurologist, again, communication wise. So when there are psychological symptoms like depression or anxiety playing a role, or apathy, again, having a conversation with the neurologist and or psychiatrist, thinking about psychotherapy, mindful awareness, meditation, and yoga have been shown to help. Support groups for the person with Parkinson's really can be useful. Sometimes people need medications for depression or anxiety. Sometimes, if there's somnolence during the day, sleep may need to be evaluated and adjusted. And I like the jolt, just one little thing. If you can get the person with Parkinson's to do just one little thing every day, that can sort of be one step forward to doing more activities. Sometimes with anxiety and depression, just getting out of the house, cabin fever. I think, one of the kind of most obvious examples of this is what happened with the COVID lockdowns. I mean, everybody was telling me about how, oh, my person, my loved one was doing so great. They were exercising, they were doing all these things. And then all of a sudden, everything stopped and they declined so rapidly with their dementia. So just so obvious that we need connection, and of course, here I am virtually. So apologies for that. I guess I'm not putting my money where my mouth is, but, even that's a connection, right? Fresh air, getting out, listening to music, breathing techniques. These are all really important. So I know there's a talk coming up shortly about sleep, so I won't go too far into detail, just to mention that sleep-related issues are common in dementia and can exacerbate them. And that can range from not sleeping at all to sleeping 12 hours plus 4 hours of a nap. So there can be a disruption of the circadian rhythm. Sundowning, people have heard of that term. It refers to being, disoriented, especially in the evening and afternoon, or getting disoriented and trying to start the day at 3 a.m., dream wake confusion, REM sleep behavior disorder. All of these things can overlap, and potential management includes a lights out routine, curtains closed, lights off at the same time every night, curtains open in the morning, limiting naps if possible, using melatonin, and discussion with sleep specialists. Last slide. Okay. I'm so grateful for all of the care partners who are here today, either online or in person. Please take care of yourselves. I know that this is easier said than done, but all of these things that, there's so much on you guys. It's like an extra full-time job, so it's really hard to stay patient and do all of these techniques and manage all of the doctor's appointments and the PT appointments and the lunch dates when you're sleep deprived and you're stressed out and you're not taking care of yourself. So please, use your oxygen mask first. Put on your oxygen mask first before you put the oxygen mask on the person next to you on the plane. Set some boundaries, prioritize sleep, get your own exercise, do your own mindful awareness. And PCLA has 2 wonderful care partner support groups. They're both online. But if it's possible with resources, hiring help can often be really important if that's possible. So that's all I have for you guys today. Let me stop sharing. What questions do you guys have for me?
Thank you so much, Dr. Petrossian. This was absolutely fabulous. We are on a time crunch, so we're going to do one question here, and then we're going to move on to Dr. Tagliati.
Just if you could speak to the importance of wearing hearing aids for cognition for
Oh,
those
yeah.
who resistant to wearing their hearing aids.
Yes, very important. Last year I gave a similar talk about communication. We did talk about hearing then too. Yes, if people aren't hearing what you're saying, then obviously there's going to be miscommunications and frustration, and also people tend to withdraw more when they're not hearing well. So loss of hearing is an independent risk factor for developing dementia. So if you want to keep your brain cells, please wear your hearing aids. If they're not working well, talk to your audiologist. There's a lot of better options now. Unfortunately, they can sometimes be pricey. And for people who don't benefit from hearing aids, cochlear implants can be considered. So please do bear that in mind, because that can make a very big impact cognitively. Thank you for that reminder.
Great question. Thank you so much, Dr. Petrossian. Thank you for joining us remotely. Wonderful content. Thank you.
Thank you. Take care, everybody.
Up next, we have Dr. Michele Tagliati. I'm going to be speaking about Parkinson's research, the most promising avenues for 2025 and beyond.
Good morning, almost good afternoon, I guess. It's 12:34, 1, 2, 3, 4. Thank you. Let me start thanking Patrick for inviting me here today. It's always a pleasure to speak to this beautiful audience. And I put in a lot of content, so I'm going to go quickly through it, and hopefully we're going to have time for some questions and answers. Perfect. So we're saying that Parkinson's disease is the fastest growing, and with the number of patients, it also our knowledge is growing, which is a good thing, obviously, but also creates a lot of knowledge, sometimes too much to summarize in a 30 minutes lecture. Those are the clinical trials and the areas of research that are currently available for Parkinson's disease. Dozens of studies, dozens of excellent ideas. And this is the number of clinical trials that are currently available for patients with Parkinson's disease. Mind-boggling, as you can understand. So I'm going to tell you that I arbitrarily will focus on a few of those ideas. And then obviously, if you have questions, I'll be more than happy to try to answer them. But before we look at the trials, we need to answer a fundamental questions that had crippled many of these absolutely valuable research research ideas. It is, what is Parkinson's disease? This is a fundamental question that we have not answered yet. We have this classic definition that we have been telling ourselves for many years: progressive chronic neurodegenerative disease, the tremor, the rigidity, the bradykiniasias, the slow and selective loss of dopamine responsive to levodopa. All true things. I mean, nothing is wrong with that. But we know that really Parkinson's disease is not that. That's a very, very small definition of Parkinson's disease. Parkinson's disease, you've seen this iceberg in many, many forms and shapes. Parkinson's disease has many, many more symptoms that point to other parts of the brain, and to the point that many researchers now are asking the question, is Parkinson's disease really one disease with many different expressions, or there are different diseases that happen to be the same or similar presentation? And this is very important because many of the clinical trials that maybe some of you even went through over the years failed in great part because we put apples and oranges and walnuts and probably kiwis in the same basket, trying to figure out whether everything worked for all of them. And in fact, this is something important that has been emerging recently, and I want to share with you. A couple of new definitions of Parkinson's disease have been offered. And they're not the end of all, but they're very important to understand if we want to be part of clinical research. So the first one comes from a group led by Tanya Simoni and supported by the Michael J. Fox Foundation. And it's trying to define Parkinson's disease as a neuronal alpha-synuclein disease. Neuronal, of course, for neurons, alpha-synuclein for the protein that accumulates in these neurons, and basically tries to create a staging of the disease based on the presence or the absence of alpha-synuclein, most importantly, and other factors. Interestingly, we heard from Dr. Mason about the genetic importance. There is also a stage 0, which is in where there is no evidence whatsoever of alpha-synuclein or dopamine deficiency, but there is that specific synuclein gene abnormality that is positively associated with Parkinson's disease. The problem with that, in my opinion, is that the percentage of people with alpha-synuclein gene abnormality is so small, as I'll show you later, that I don't know what is the value of that in practical terms, but it's there. Then there is a stage 1 that is based on the detection of alpha-synuclein with a new test, and many of you, I'm sure, are familiar with this alpha-synuclein amplification test in the cerebrospinal fluid, which is something, in all honesty, not very common and very easily obtained. You need to do a spinal tap, basically. But again, these are research definitions. This is not something that your doctor will do on a daily basis. So in the future, to participate to certain trial for Parkinson's disease, many will ask you to do this test in order to confirm that you indeed have alpha-synuclein abnormalities in your brain. The problem with that, just to let you know, is that not every patient with Parkinson's disease have that abnormality. For example, those who carry the LRRK2 mutations that Dr. Mason discussed earlier may not have that, although it's difficult to say that they don't have Parkinson's disease. They have all the symptoms of what we call Parkinson's disease. So just to give you an idea that we're making progress, but it's still not perfect progress. And then obviously we can look at dopaminergic dysfunction, most likely with the DAT scan. So there are people that will have only the synuclein, people that have the synuclein and the DAT scan. That will be stage 1B. Then one of the most interesting part of the story is that stage 2 is when you start having symptoms, but not the symptoms that we call Parkinson's disease today. But this enlarges the symptom of Parkinson's disease to the so-called premotor symptoms, the acting out of the dreams, the constipation that we heard about brilliantly earlier, the anxiety, loss of smell. And those are, if you have a dysfunction of synuclein in your cerebrospinal fluid, and you don't have technically what we call Parkinson today, but you're just acting out your dreams, you are a stage 1A, sorry, 2A. So you have Parkinson's disease. So we are enlarging the concept of Parkinson's disease to symptoms that until yesterday, people will never worry about. And why we do that? Because that, as you can imagine, is probably one of the key moments in which we can act most effectively to slow down, to change the progression of the disease. So we're really changing the definition of Parkinson's disease from something only neurological to a disease that starts many years earlier with symptoms that are not only neurological. And then stage 3 to 6 is more what we currently call Parkinson's disease with the neurological impairment. There is another classification, just to keep things confusing, that has been proposed by another group, led by Dr. Tony Lang, that tried to keep into consideration, again, the fact that it's probably difficult to pin down just one type of Parkinson's disease. So try to capture the heterogeneity of Parkinson's disease and to help research sort of collecting participants that look like each other. And they look at 3 key components. One is the deposition of alpha-synuclein, one is the neurodegeneration, and one is the gene. And without getting into too many details, in addition to the cerebrospinal fluid, they also accept the skin deposition of alpha-synuclein is much easier to obtain with a skin biopsy as evidence of alpha-synucleinopathy. They obviously look at the DAS scan, but also at the PET scan, which some centers do, but also the MIBG, which is a heart study. So it's kind of sort of opens up a little bit the terms of the definition, and then the genetic part is very complex because we have penetrant, predisposing genes, intermediate, low predisposing. Again, gives you an idea of the complexity that researchers have to face when we try to decide who is a good candidate for a clinical trial or not. So that said, I arbitrarily try to focus on a group of lines of research that I believe might be relevant. And disclosures, these happen to be the type of research that we are running at Cedars-Sinai. So it's not to bring you to Cedars, but it's to speak about something that I have some background and some title to describe. So initially, let's look at disease that, trials that, or ideas that target disease modification. So an attempt to slow down or stop Parkinson's disease somehow. The first one is genetics. We heard earlier from Dr. Mason a lot about genetics, a couple of concepts. So this is a big study. It's called the Rostock Parkinson disease study that examined the blood of over, you see, 12,000 patients with Parkinson's disease. We contributed to that, and they found that only 12% or even less than 15% of patients with Parkinson have a recognizable genetic variant or mutation. I don't like mutation, sounds something, you know, like a mutant, like an alien. It's a variant, really. It's perfectly compatible with normal life, at least until your 60s or your 70s. But interesting, this is way too small to read, and I apologize for that. There are a bunch of other variants of uncertain significance, VUS. In other words, people also carry another bunch of variants that we're still not sure what they really mean. They've been associated with other diseases, but they may be relevant even if in a smaller part also for Parkinson's disease. And what I find very interesting is that even patients that did not have any of these variants, still a quarter of them had a positive family history, or at least they claimed that someone in their family also had Parkinson's disease. So there is so much more we need to learn about the influence of genetics. But as we heard already, the fact of having or not having a variance already throws an interesting wrench into our ability to classify and to divide Parkinson's disease, because if you have the LRRK2 mutant or the GBA mutant, you see you go to different type of benign or not benign. Dr. Mason mentioned that earlier. So it adds complexity to what we're trying to do. And look at this. This is just the number of genetic loci associated. We already heard about GPA. So let's focus on what we're trying to do. So this is interesting. GBA and LRRK2 results in protein or enzymatic defect that are opposite. GBA, or glucocerebrosidase, is an enzyme that if you have a GBA mutation, you tend to produce in a smaller amount. So it becomes insufficient. You don't have enough. And so you need to do something to potentiate, to help these enzymes work better. And there are, interesting, the GBA mutation is also a cause of another disease. It's called Gaucher disease. And there are treatments for that. But however, it doesn't really look that they will be able to help with Parkinson's disease because they don't cross the blood-brain barrier, so they can't get to the brain easily. So that would have been nice. That would have been easy, but it's not so simple, unfortunately. So there are a number of attempts, and the one that is probably more accessible today, and I have some of my patients that do it spontaneously, is to take Ambroxol. Ambroxol is a cough medication. It's very easy. There's only one problem, that is not available in the United States. Yeah, but it's currently available in Europe. You can get it without even a prescription in a pharmacy in Germany or in Italy or in Switzerland. And in fact, many of my patients tend to go there and they claim that it helps a lot. There are studies, preliminary study, that showed an improvement in EUPDRS and non-motor scale, quite interesting. So that is, to me, something that has promise, at least empirically, because again, I personally follow a number of patients that claim that once they started taking, of course, it could be a big placebo effect, Ambroxol, they felt better. So write it down. That will come to the United States at some point, but it's not available in the pharmacies now. Then there are a number of more sort of scientifically complicated trials that are currently ongoing, some easier than others. This one requires a intracisternally, which means inside your cerebrospinal fluid application, that are ongoing for GBA. But the take-home message is that, and to answer some of the questions about should I get tested, I truly believe that we'll get to a point not far from today in which every single patient with Parkinson will get tested for genetics to see whether we can personalize their treatment with one of these efforts. This is the other common or relatively common variant, which is LAR2 genes, which is much more complicated because it also predisposed to other problems, like, for example, Crohn's. And the LAR2, interestingly, if you have a variant, you end up having too much of that enzyme. So you kind of need to slow it down. And there is a so-called toxic gain of function. Sometimes too much of a good thing is not a good thing. And the attempt to treat LAR2, there are two. One is the Denali inhibitor that try to reduce the phosphorylated LAR2, the abnormal LAR2 in the blood, and is currently a focus of at least a couple of clinical trials. One is called LUMA. Some of you might be familiar with that. Another one is in preparation, and Denali is the sponsor. And then there is a very interesting scientifically trial in which there is an antisense oligonucleotide. What does that mean? It's basically a piece of DNA that tries to block the part of your DNA that produced the wrong gene. You can intuitively understand that many of these efforts are very interesting scientifically, but we need to take them really with a grain of salt, because these are genes that don't just produce the wrong protein for the brain, but they produce the proteins that goes everywhere else in the body, and you can't afford to stop completely producing it. So very interesting ideas that still have to be proven, not only effective, but safe, which is the most important part of the story. Very good. Another target that I find extremely interesting is inflammation. What is the role of inflammation? What is Parkinson inflammatory disorder? Probably yes. And there are a number of evidence that brings us to this definition. First of all, age, which until proven otherwise is the most important risk factor for Parkinson's disease, is associated to alteration of our immune system, which is something that we all know. Trust me, I am part of that group myself. You tend to be more prone to getting infections as you grow older. And that's because there are changes in your immune system, and you start reducing your immune cells, and those immune cells start to become less effective. And also, you have environmental stressors that start to accumulate over your lifetime: viral, bacterial exposure, pesticides, diet. We discussed earlier the role of the gut microbiota, that are all associated one way or another with an increased risk of getting Parkinson's disease. And in fact, here is an ideal. See the dysbiosis that we discussed earlier today, increased level of pro-inflammatory proteins. The immune system starts to become weaker. The blood-brain barrier starts to become more leaky. And last but not least, you have what we call neuroinflammation. You start having literally inflammatory events slowly going at your brain. And there are a number of evidence. For example, patients that for some reason use anti-inflammatories in their life, maybe they have arthritis or they have other inflammatory disease, they tend to have less Parkinson over time. So taking aspirin and similar medication seems to be good. Please don't go out there and start taking aspirin. These are all retrospective data that try to support the role of inflammation. A genome-wide association studies is one of those more complex and expensive studies that Dr. Mason discussed today, showed that patients that have Parkinson's disease and autoimmune diseases seems to have identical genes, which might be predisposing them to have one or the other. LRRK2, which gene variations that we just reviewed, increase risk of developing Parkinson, and interestingly, inflammatory diseases like Crohn's disease. There are families in which one sibling has Crohn and the other sibling has Parkinson's disease. The father has Crohn's and the children have Parkinson's disease. They seem to have a lot in common. And in fact, patients with autoimmune diseases like multiple sclerosis and others have an increased risk of developing Parkinson's disease. More than that, if you have an inflammatory disorder and you're treated for that, for example, with anti-TNF or anti-interleukin 17 treatment, they decrease the risk to having Parkinson's disease. So there is a very close association to inflammatory diseases and Parkinson's disease. We can also see that patients that, this is a study that Camille Malat at Cedars-Sinai has performed. We have looked at 1,200 patients with Parkinson's disease, and we asked them what medication they were on, as we ask obviously to every one of our patients. And those who were on anti-inflammatories, adrenergic blockers, so beta blockers, and cholesterol medications, all of them have anti-inflammatory roles. They tend to develop Parkinson's disease 10 years later than everybody else that were not exposed to those medications. Again, observations which don't make a clear causation link, but all these observations seems to tell us that if we pay more attention to these inflammatory processes, we might change the course of neurodegeneration. And there are a number of biomarkers, because we can't just look at the symptoms. Sometimes neuroinflammation for years may have no symptoms whatsoever. You can be a perfectly normal person and have a brewing inflammatory process. But you see patients with Parkinson's disease have increased, many inflammatory markers increased. And also, if you do a specific type of PET scan, TSPO PET scans, you can see the Parkinson patients just diagnosed with Parkinson, so very mild Parkinson, already have abnormal binding in many areas of the brain. So the field is ready. We have the idea, we have the markers that we can follow. Do we have something to use? The answer is yes, but you have to be careful because when you use anti-inflammatory medication, you reduce the ability of your immune system to react to infections. So the risk of immunosuppression can be very high. You hear the commercials on the radio and TV for all these medications for Crohn's, for ulcerative colitis, and they always say, tell your doctor if you're getting a vaccination, tell your doctors if you have an infection, risk of tuberculosis, right? It's pretty scary. They're all true. The risk is small, but it's there. And also, probably it will take more than one drug. The one drug cures all is probably a dream that we will never achieve. We will need multiple drugs to affect multiple risk factors for Parkinson. But let's look at a couple of ideas. One is the GLP-1 agonist. Did anybody know Ozempic here? Yes, we're in Los Angeles, right? We know Ozempic. But seriously speaking, GLP-1 is a medication that is extremely helpful for diabetes. My endocrinologist friend love GLP-1 agonists, Ozempic, Victoza, exenatide, you name them. But this medication, in addition to prevent diabetes or to cure diabetes or to treat, I should say, diabetes, they can prevent inflammation. And that's probably one of their major roles. And in fact, we have been testing GLP-1 agonists in Parkinson's disease. And there have been 3 medications tested: exenatide, liraglutide. We tested it at Cedars, lixisenatide in a French study. And we all found some interesting improvement either in motor function, or we found them in non-motor function and quality of life at Cedars. Again, please don't go out there and tell your doctor to give you GLP-1 agonists because they have a number of side effects, especially on your GI. But definitely these are interesting ideas, because why a diabetes medication should help someone with Parkinson's disease unless the common thread is the anti-inflammatory action of this medication. And this is another concept that is very new. It's the so-called NLRP3 inflammasome. Is a protein which is in the brain, which when it assembles, it starts to activate inflammation. Now, inflammation, again, anti-inflammatory action is very important, but it may start creating too much inflammation and create problems. So when activated, this NLRP3 inflammasome release all these cytokines and can lead to inflammatory disorders. So we have been recently testing in patients with early Parkinson's disease this complicated number RO is a Roche compound called Selnoflast. We just finished an initial testing. I'm not authorized to share the results, but I've been told that they are encouraging. So keep your attention tuned to this potential anti-inflammatory medication. So neuroinflammation, insulin resistance, abnormal stress response are probably working together to lead to neurodegeneration. But let's go back to aging to introduce a new concept. It's the one of senescent cells. Who has ever heard of senescent cells? One person, which is very little, right? Because nobody talks about senescent cells in neurology. This is a very, very well-studied topic in other diseases like cancer or diabetes, but very little in neurology and neurodegeneration. What are senescent cells? Senescent basically means aging. These are cells that naturally, under aging, tends to stop dividing, stop doing what they're normally doing, but they don't die. They kind of sit there. And when they sit there, they may change their conformation and starts producing cytokines, inflammatory processes. And they can be positive. They can do important roles, like, for example, wound healing, or they can help development. But sometimes they turn into monsters, and they can literally produce what we call aging. The stiffness, your gray hair, the skin that becomes so wrinkly, it's been associated with the action of the senescent cells. And we are starting to study whether this could be something that is at the base of Parkinson's disease, because we all talk about what may cause Parkinson, but we never know where it starts from, where all this inflammation or whatever else we decide could cause Parkinson's disease come from. It looks always to come from Zeus with a big lightning that hits our brain. But this might be something normal that we all experience, but that, again, from some genetic predispositions or due to the number of viral infections or exposure to trauma or to toxic elements, might increase our chance to having senescent cells, then cause aging or cancer or other disease. And the good news is that there are senolytics, so there are compounds out there that can get rid of senescent cells. And they've been tested in many other disorders. The only one that get a little bit closer to Parkinson is Alzheimer's. There are very, very initial studies at Cedars that have shown that some of these compounds are relatively safe. We need to see whether they are actually effective. But you see how many studies not related to forget Parkinson, but just neurological disorders. So this is a fairly advanced science, but not in neurology or neurodegeneration. And we have to be very careful, however, because eliminating the senescent cells can cause, problems to other physiological function that these cells might have. So it's something that I personally find extremely exciting, but still very, very early in its development. So quickly, let's jump forward to the advanced stages and to deep brain stimulation. As many of you know, this is something that I've been working on for many years. And deep brain stimulation is evolving. It's an amazing technology. It works very well. I'm sure that someone in this room has deep brain stimulation implanted and benefited from that. But what is going on with deep brain stimulation? So we are having a number of technological improvements that are currently tested in very, very interesting studies. We, for example, have new 7 Tesla MRI that can give you incredibly precise pictures of our brain. And in addition to that, we can now trace not just the nuclei, where the cells are, but also how the cells connect to each other, which is probably even more important when you try to insert electrical pulses, right? When you start stimulating the brain, that electricity goes pretty much everywhere in the brain. So it's very important to know exactly which highway you're touching. One thing is you get on 405. One thing is you get on 110. You go to different places. So you want to hit the right highway. And then there is adaptive simulation. One of the limitations of DBS is that, as effective as it is, we throw in some stimulation, and that is 24/7, no matter how you're doing. You're sleeping, you're awake, you're running, you're sitting. It's always the same. Boom, 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 boom. And we know that that's not how it works. I promise I have two more things to say. I know that I'm long, but okay, give me 2 more minutes. So the other issue is artificial intelligence. Deep brain stimulation is by definition something very complicated and very complex. That's why has not taken as wide as a practice as we hoped, because it's difficult to control. And so artificial intelligence, as I'll show you quickly, will help us. So what is adaptive stimulation? So we know that the brain of patient with Parkinson has a specific abnormality that has an increased beta power in the subthalamic nucleus. And the good news is that when you take levodopa or similar medication, it goes away. Now we can, with the same electrodes that we implant to stimulate the brain, we can also record, we can sense this abnormal activity. And the good news that similar to levodopa, deep brain stimulation can also eliminate that abnormal activity. So we believe that we can know when the brain is functioning normally or similar to normal, and when instead is not functioning normally. So you can make the next step and see that if we can tell, if we have a device that can adapt the amount of stimulation based on how many of those abnormal activations they can record, we can tailor the stimulation to what the brain really needs. So it's not a 24/7 bombardment, but it's a much more natural activity. And this is not just something that, you know, will save battery life, but I don't know if it's easy to see, but patients with continued deep brain stimulation very often develop problems walking, which the adaptive nature of the stimulation seems to resolve. This is all very initial. That's why I put it in the, you know, future interest, but it's definitely something that can bring DBS to the next stage to act specifically when the brain needs it. And this is the key slides for the adaptive, artificial intelligence. Do you see these are all the different variables that we need to control when we stimulate the brain? And if we then now introduce even adaptive DBS, this will multiply exponentially. And so definitely AI will be extremely helpful. And there is already one program available that will help us. You see, this is a typical patient with some tremor. You can take off the audio if you want. And you see, when we see these, we try to stimulate the brain in a way this we can visualize and try to direct our stimulation to the appropriate nucleus in order to obtain our effect on the tremor. But that takes a lot of time, a lot of trial and error. And to have a system that can, just by telling him, I want to stimulate the subthalamic nucleus and I want to avoid to stimulate the motor fibers, bang, he gives me exactly what I need to stimulate. It's something that only AI can do, and is something that we are testing currently in order, number one, to optimize without using hours and hours of testing, but also allow centers and physicians that are not necessarily expert like Dr. Mason or myself to immediately give their patients the best possible optimal stimulation. And this was my last slides. So fastest growing among neurological diseases. We have reviewed the new biological classification. There are multiple fields of research. We look at genetics, inflammation, and the future of DBS and artificial intelligence. I just want to thank my team at Cedars-Sinai. It takes a village to take good care of our population of patients with Parkinson. Thank you for your patience and for your attention.
Thank you so much, Dr. Tagliati. I'd like to open it up for one question. Does anybody have a question?
Oh, again, very good question. So the question is Ambroxol versus carbidopa levodopa. Ambroxol would be with carbidopa-levodopa, not as opposed to carbidopa-levodopa. I don't think that nothing as of today, and probably for the foreseeable future, will beat carbidopa-levodopa in terms of symptomatic help of your motor symptoms. What all these trials seem to look for is something that, in addition to the symptomatic, to the benefit of levodopa, we can do to slow down the progression of the disease, because levodopa makes you feel great today and for the next 10, 20 years, but it doesn't seem to do much to the progression of the disease. We are trying to do something that, in addition of improving your symptoms, keep them there for the rest of your life. This is really the vision. And so treatment like Ambroxol try to counteract possible contributing factors, like, you know, in the GBA case, the reduction of the glucocerebrosidase enzymes with Ambroxol seems to help working better. And so they're parallel, they're friends. I specify one thing, and I want to say it again. The one treatment cure it all is not a paradigm that we probably ever achieve, and it's probably not even reasonable. Look at cancer. People with cancer don't take just one drug. They take a number of medications. Diabetes, even hypertension, right? High blood pressure. Sometimes you need to take 2 or 3. I don't think that finding the cure for Parkinson will need to find the magic bullet. We'll probably need to find 5 or 6 magic bullets, which might be different for each and one of you, because each and one of you, as we said at the beginning, might have a slightly different rendition of the same disease, or maybe different diseases altogether. This is the challenge for us, and obviously what slows down our ability to do what we all want, which is to get rid of this problem. Thank you. Thank you again for your attention.
Wonderful. Thank you. Next we have up Dr. Adrienne Keener.
Right. Hello, everyone, and thank you for joining us today. And thank you for hanging in there to the end. I have the pleasure of talking to you all about sleep. So hopefully I don't put you to sleep as we wrap up the program today. Okay, so I think you've heard a lot today about Parkinson's really being a multi-system disease. So we've talked about motor symptoms, non-motor symptoms. We're going to focus today on the non-motor symptom of sleep. The iceberg image you've seen also repeatedly, and I just will emphasize here how important it is to emphasize the treatment of non-motor symptoms for your quality of life. And sleep is one of those that we don't often think much about because we're sleeping until we're not. So the average person with Parkinson's experiences up to 12 non-motor symptoms. And the burden of these symptoms increase as the disease progresses. We know that this is associated with increased morbidity in patients with Parkinson's disease, placement in nursing homes. So these non-motor symptoms really are critical. So why do we need to sleep? Well, sleep has a very important biologic function to detoxify and replenish our bodies and minds. And up to 90% of people with Parkinson's disease have sleep problems. I would say pretty much everyone with Parkinson's has sleep problems. The other 10% of people maybe didn't understand the question in the survey, because I don't think I've ever met a patient who doesn't have an issue with their sleep. So we're going to review some of the more common sleep issues in people with Parkinson's, including excessive daytime sleepiness or fatigue, insomnia, which can manifest as difficulty falling asleep and also difficulty staying asleep, REM sleep behavior disorder, and restless leg syndrome. So very briefly, I'll just highlight here, there are gender differences in the way that people experience Parkinson's disease, and that includes sleep symptoms. So studies have found that symptoms of fatigue, insomnia, and restless leg syndrome seem to be more common in women with Parkinson's disease, while complaints of daytime sleepiness and REM sleep behavior disorder seem to be more common in men with Parkinson's disease. And among those with REM sleep behavior disorder, interestingly, the dream content can also be different between women and men. And so I think just important to note that even if you have the same diagnosis, the same sleep disorder with Parkinson's, you may experience it differently than someone sitting next to you here today. All right, so starting with daytime sleepiness and fatigue, a complaint in over 50% of patients with Parkinson's disease, and for up to a third of patients, this can actually be their most disabling symptom. So why do you feel so tired? Well, we know that Parkinson's disease, in addition to impacting dopamine, impacts your arousal systems in the brain, can also disrupt your circadian rhythm. So this can lead to a vicious cycle where you find yourself sleeping more during the day and then awake more at night. And if that goes on for a few days in a row, it can be really difficult to get back on schedule. We also know that medications that we prescribe can cause side effects that make you sleepy. Depression can also manifest with fatigue as a symptom. And we know that depression is very common among patients with Parkinson's disease. And finally, some of the other nocturnal disturbances we're going to talk about tonight can also make you sleepy during the day. So if you're not sleeping well at night, of course, you're going to be more tired and fatigued during the day. So how do we manage sleepiness? I'll start by saying avoid driving if you're sleepy. But one of the first things we look at is medications that might be the culprit. So if we can stop a medication that's causing you to be more sleepy, that's an easy fix. So levodopa very rarely can cause sedation as a side effect. At higher doses, probably more common. Dopamine agonists can definitely be more sedating and can actually cause a phenomenon that we call sleep attacks, where in the middle of eating your meal, you sort of doze right off, right? And finally, anticholinergic medications, which are sometimes used to treat tremor in Parkinson's disease, can also be sedating. And these are just a list of the movement disorders medications you might be prescribed, not to mention other medications you might be on for other reasons that can also cause sedation as a side effect. Next, we can try to sort of reset that sleep-wake cycle, staying awake during the day, trying to engage in more sort of physically or mentally, intellectually, socially stimulating activities so that you're up more during the day, promoting better sleep at night. Light exposure can be really key. So getting morning sunshine, natural light, can help reset your brain circadian rhythms. Our brains are very sensitive to light, and you'll hear a bit more about that later when we talk about insomnia. And lastly, sometimes this symptom can be really difficult to treat. We've optimized everything else we can. We're still really sleepy during the day. And that's when we might reach for a stimulant-like medication like modafinil or methylphenidate. And next, we'll talk more about sort of addressing those sleep disturbances, right? So how can we get you sleeping your best at night so that you can be as alert and awake as possible during the day? So let's start with insomnia. So as I said, this can be either difficulty initiating sleep or also difficulty maintaining sleep. And it's actually that sleep maintenance that's the most common complaint among Parkinson's patients. So they might say, I have no problem getting to sleep, but then I wake up after 3 hours. I toss and turn for a couple more hours. Maybe I'll doze back off for an hour or two. It's just really fragmented, really difficult to maintain sleep. This is a very common problem even in the general population. But in Parkinson's disease, up to 80% of people report insomnia. There are some common risk factors, depression and anxiety, so mood disorders. Certain medications can contribute to insomnia. And also this seems to be more common with longer disease duration. So insomnia is often multifactorial in Parkinson's disease. So motor symptoms at night can contribute to insomnia. So we're treating you with your levodopa throughout the day to try to control your motor symptoms. But then overnight, sometimes your medications might wear off. And we know that the more you might be developing motor fluctuations during the day, the more you might be experiencing that wearing off overnight. So this can manifest as, you wake up to use the restroom, your tremors come back out, and then that keeps you from getting back to sleep. Or you're having muscle stiffness or even dystonia in bed, making it very difficult to maneuver and get comfortable at night. Nocturia, this is having to wake up to go pee. So if this is happening very frequently overnight, then obviously your sleep is going to be very disrupted. Nocturnal pain is also a common phenomenon in Parkinson's, We don't often think about sensory symptoms in Parkinson's disease, but pain certainly can keep you from sleeping well at night. REM sleep behavior disorder we'll touch on in a moment, but that's vivid dreamings that you might act out at night. Sleep apnea. So if you have sleep apnea, that can also disrupt your sleep at night. I mentioned mood disturbances that can often manifest with insomnia. And if you have hallucinations, psychosis, sort of agitation or sundowning that happens in the evenings, that can also make it really difficult to get to sleep, as Dr. Petrossian highlighted earlier. And finally, you might just not be a good sleeper. And we're going to talk a little bit about sleep hygiene in a moment. So when we approach treating insomnia, we really want to break down, which of these factors is causing your insomnia. And it might be many of them warranting unique treatments for each of those symptoms. So if you're having motor symptoms overnight, we want to treat those. There are long-acting dopaminergic medications we can give at bedtime. And then there are some newer, more advanced therapies that have been shown to have a positive impact on sleep. So continuous levodopa formulations, including the intrajejunal pump, and most recently approved the subcutaneous pump. Deep brain stimulation also associated with improvement in sleep. Nocturia, so if you're getting up to pee a lot at night, overactive bladder or urinary urgency and frequency are very common in Parkinson's disease. We can try some behavioral strategies to address this, limiting how much you're drinking liquids in the evenings. Oftentimes our urologists are our good friends and can also prescribe medications to help decrease this symptom. And finally, if you have mood symptoms that are untreated, depression, anxiety, addressing those symptoms can also help improve your insomnia. There are also some non-pharmacologic treatments for primary insomnia. So when we've addressed all those other issues, so cognitive behavioral therapy for insomnia, or CBTI, is a form of psychotherapy specifically for insomnia. There have been some studies looking at this approach in people with Parkinson's disease and showing that it is efficacious. Bright light therapy. So more than just getting out for a walk in the sunshine in the morning, but actually intentionally exposing yourself to bright lights to help reset that circadian rhythm may help promote sleep later in the evening. Exercising. So exercising during the day, sort of physically getting yourself tired can help promote better sleep at night. There are also some, mindfulness or meditative approaches that can really help you wind down and get to sleep. And lastly, let's talk about sleep hygiene. So sleep hygiene is sort of a set of best practices. How can you best promote your sleep and help yourself stay asleep longer? So first is, the brain loves a routine. You got to go to bed and wake up at the same time every day. So we are not teenagers sleeping in on the weekends. Our brains really love, this is my bedtime, this is my wake time. That morning sunlight, again, especially pairing it with some exercise, I think can do wonders for setting your daily routine. We want to avoid stimulants like caffeine too late in the day. We all know that will definitely disrupt your sleep. That being said, I think a nice cup of coffee in the morning is certainly permitted, right? And might actually help promote wakefulness when you're first getting up for the day. We want to also maybe avoid, stressful topics, activities right before bed. So, if the news is stressing you out, don't watch it right before you're going to go to bed. Avoid napping during the day. So if you are going to have a brief nap, it should be brief, a 20, 30-minute power nap. But if you're sleeping for 2-3 hours during the day, that is going to impact your ability to sleep well at night. Also, we want to avoid eating or drinking too close to bedtime. So having a heavy meal in particular, especially if there's alcohol involved, will disrupt your sleep. And lastly, I mentioned earlier, our brains are very sensitive to light and screens. That blue light from TV, cell phone, from your computer, your tablet, that light sort of tricks your brain into thinking it's morning time. And so then your circadian rhythm gets further disrupted. You have a hard time getting to sleep. So if you can try to put aside those screens for an hour before bed and have a nice wind down routine, that's another good practice. And having that routine really helps cue your brain that it's time for sleep. Your brain is smart. It will recognize, oh, hey, we're doing the same thing every evening right before we go to bed. So it sort of will learn to anticipate that sleep is coming. This should be a calming routine, something like listening to some quiet or calm music, reading a book, journaling perhaps, listening to a guided meditation, but it should be sort of a calming routine that you do every single night, brush your teeth and go to bed. Also, your sleeping environment is quite important. So you should be sleeping in a dark place. It should be cool. The American Association of Sleep Medicine actually recommends 65 degrees. So our bodies actually sleep better in cool environments. Think about your bedding. Is your mattress comfortable or appropriate for you? Is your, the sheets and the covers, are you getting really tangled up and that's waking you up? I heard a patient tell me that they got silk pajamas and that helped them sleep a lot better because they sort of were sliding more easily, under the covers. Do you need a humidifier in your, it gets really dry here in the winter months. And sometimes a humidifier can just make it more comfortable to breathe at night. There's an approach in the sleep literature called reconditioning, and this is sort of retraining yourself that the bed is only for certain tasks, only for sleep and intimacy. Don't be working in bed. If you're laying in bed and you're not sleeping, get up and go to another room and do something else, and then come back to bed when you're actually ready to sleep. Try your best to minimize looking at the clock if you wake up in the middle of the night, right? That clock watching can really make sleep feel like torture, just staring at the clock. If you do have frequent nocturia and mobility is a challenge at night, something like putting a commode at your bedside just to make that safer and easier can be helpful. And again, exercise is great for many reasons, but not too late in the day. So if you're saving your exercise for the hour before you go to bed, again, you're sort of turning everything on, ramping everything up. It's best if you can time your exercise earlier in the day. Okay. Finally, we've tried all of these approaches. We do sometimes reach for medications. We'll talk a moment about melatonin and REM sleep behavior disorder. There's some other medications that can be used for sleep, though I will say they have pretty limited data. And some of them are actually recommended sort of to avoid in people who are over 65 because of side effects. Pimavanserin, Nuplazid, which we sometimes use to treat Parkinson's related psychosis, can also perhaps improve sleep in those patients. And lastly, I will say a lot of over-the-counter sleep aids include a medication called diphenhydramine, which is the generic for Benadryl. Avoid, avoid, avoid. This medication has a lot of anticholinergic side effects. It might knock you out, but it is not a safe or recommended sleep aid, especially in the long term. So let's talk about REM sleep behavior disorder next. So normally during rapid eye movement or REM sleep, there's sort of a switch in our brain that prevents us from acting out our dreams. In REM sleep behavior disorder, that switch is a little faulty, and so that inhibition of motor activity is lost. We end up acting out our dreams. This can manifest as talking, laughing, shouting, gesturing, kicking, punching, even sitting up or leaping out of bed. This can occur in 25 to 50% of patients with Parkinson's disease and can even precede the onset of the motor symptoms by decades. And in fact, it's one of our most closely associated prodromal sort of non-motor symptoms. So people who develop REM sleep behavior disorder, we know if we follow them over time long enough, they will eventually likely get an alpha-synucleinopathy like Parkinson's disease. REM sleep behavior disorder, again, several studies have found more common in men with Parkinson's disease. Also more common if you also are in an advanced stage of Parkinson's disease and also experiencing some cognitive impairment or hallucinations. Antidepressant use can also contribute to REM sleep behavior disorder, though this is not a reason usually to discontinue your antidepressant, and then alcohol use. So the gold standard for diagnosing REM sleep behavior disorder is an overnight sleep study where they hook you up, they're measuring your motor activity, they're measuring your brain waves, they're measuring your oxygen in a sleep lab. For many people, that's quite a cumbersome test and maybe not even feasible. So there are very good screening questionnaires that can help us identify patients who most likely do have REM sleep behavior disorder. And there's even a one-item version of this questionnaire. And the question is, have you ever been told or suspected yourself that you seem to act out your dreams while asleep? And we give some examples of what that might look like. So you can, ask yourself or your loved one that question. And if the answer is yes, maybe talk to your doctor about this symptom. So the goal of treatment in REM sleep behavior disorder is to minimize those disruptive dreams, any associated injuries, or sleep disruptions that might occur as a result. And this is for the person with Parkinson's disease and also for their bed partner who might be a victim of their kicking or punching in the night. So education and counseling our patients and their partners around REM sleep behavior disorder is important for safety. If possible, we try to discontinue any medications that might be exacerbating this. It's really important if you have sleep apnea to also treat that, because we know that if you have untreated sleep apnea, your REM sleep behavior disorder will be worse. Melatonin is a very safe and effective treatment, and I think for most of us, the first line medication we use for REM sleep behavior disorder. Low-dose clonazepam, which is a benzodiazepine, is extremely effective for this condition, but also carries its own risks as a benzodiazepine. So we usually reserve that as the second choice if melatonin is not sufficient. We'll wrap up here our sleep section with restless leg syndrome. So restless leg syndrome is characterized by an urge to move that's often quite uncomfortable. People can describe this sensation in a variety of ways, but it's sort of can be a pain, can be a sort of wriggly, sort of, creepy crawlies kind of feeling that's associated with the urge to move. Symptoms are occurring exclusively at rest. They're worse in the evening, usually exclusively in the evening at the beginning at least, and are relieved by movement. So getting up and moving around helps alleviate the symptoms. So you can imagine if you are laying down to go to bed at night and then your legs start to experience this uncomfortable sensation, you have to get up, walk around, that's going to make it difficult for you to fall asleep. Restless leg syndrome often co-occurs with something else, another acronym, PLMS. This is periodic leg movements of sleep, and these are jerking movements that happen when you're asleep. And if those are very severe, they can also awaken you or your bed partner. So how do we treat RLS? We know there are several factors that can exacerbate this, including caffeine, nicotine, alcohol. Certain medications can also exacerbate restless leg syndrome. There are a lot of behavioral or lifestyle approaches that can help improve these symptoms. Exercise, again, plays a big role here, including stretching, yoga, tai chi, applying warm or cool packs to sort of distract that sensation, massage, a warm bath before bed. There are even some devices that have been developed to try to help with this symptom, like a foot wrap that applies some pressure on the ball of your foot, a vibrating pad that you can put under your legs. Some of these have been suggested to help with restless leg syndrome. Finally, if you have this symptom, you should check your iron stores. In many people with restless leg syndrome, their iron stores are low, and so repleting their iron levels can help with the symptoms. And then there are medications as listed here, the gabapentin, pregabalin, dopamine agonists, and if very refractory, opioids can be effective treatments for restless leg syndrome. So I'll wrap up here with a few take-home points. Sleep issues are very, very common in Parkinson's disease, and we know that getting a good night's sleep is critical to your health and overall well-being. Identifying contributions to poor sleep from motor symptoms as well as other non-motor symptoms is important so that we can give you targeted treatments addressing those symptoms. And finally, as with a lot of Parkinson's disease care, treatment really must be individualized and often involves sort of lifestyle or behavioral non-pharmacologic approaches in addition to some medications. So with that, I will take any questions and thank you again for hanging in there.
Thank you so much, Dr. Keener. Fabulous. Do we have some questions?
Thank you. Thank you. So I suffer from nocturia and I do all the hygiene that they tell me to do.
Yeah.
And I still get up 3 to 4 times at night. And I'm wondering if it's Parkinson's related or it's just overactive bladder related or something else.
Yeah, so overactive bladder can be due to Parkinson's. So we know that Parkinson's can impact your autonomic nervous system, and that includes your bowels, your bladder, even temperature dysregulation, which can also affect sleep that I didn't mention specifically here. So if you have overactive bladder, it likely is directly related to the impact of Parkinson's on the nerves that control your bladder function. That being said, we don't have a Parkinson's specific treatment for overactive bladder, but medications can be used to help with that overactive bladder. And then, prostate issues are also very common as people get older. And so addressing those symptoms with your urologist is also important. Dr. Tagliati has a comment.
Yeah. So one important question that you need to ask yourself is, am I going to the bathroom every hours during the day or only during the night? If it's only during the night, you don't have an overactive bladder. You have an interrupted sleep. Because what we will do, even without having Parkinson's disease, every time we wake up during the night is going to be to fill our bladder. If you think about it, all your life, the first thing you did in the morning is going to the bathroom. So unless we believe that human beings wake up in the morning to pee, which obviously is not the case, that's what we do when we wake up. So if you go to the bathroom every hour during the day, you have overactive bladder. If you wake up and you go to pee only during the night, you have fragmented sleep and you need to take care of your sleep. If you sleep better, you will wake up less and you will go to the bathroom less. So this is, in my view, at least the difference.
Yeah.
And unfortunately, every time someone goes to the bathroom during the night, is labeled as overactive bladder. And they take whatever medication for overactive bladder, and then they come back to me and say, man, I didn't do much because the problem is waking up. So this is the rule of thumb, that the question that you need to ask yourself or ask your partner is that frequency during the day, independent of how much you drink or you don't drink and things like that. So it's a very important problem. It's probably one of the most important problems the patient with Parkinson face, which is not sleeping well. If you may know that not everybody has tremors, for example, only 60%, maybe 70% of patients with Parkinson have tremor, but how many don't sleep well at night? I want to take a guess. 99%, meaning all of them. And in fact, yeah. I sometimes define Parkinson's disease as a sleep disorder and not a movement disorder. So that's very important. It's something that disrupts the quality of life of virtually every single patient with Parkinson. And that's one rule of thumb that can help you decide whether you want to knock at Dr. Keener, say, hey, I need something to sleep better, or knock at the urologist's door and say, I need to regulate my bladder.
I think you're right. Sometimes we'll try to tease that out, trying to ask, like, are you waking up because you have to pee, or do you wake up and then figure you might as well go pee, right?
So I have the what do they call it, RBD or
RBD.
Yeah. Like I just had one last night, right? So what is the role of when you take the last carbidopa dose that causes RBD or not?
Yeah, so that's a good question. So I think, sometimes we feel that taking like too much immediate release levodopa late at night can perhaps be stimulating, though there are also some studies that show that levodopa might actually decrease the amount of REM sleep that you're having. So in doing that, then you might have less RBD. But I do think it is worth paying attention to sort of the interplay between your, dopaminergic meds and how much dream enactment you're having. And so, if you haven't sort of taken a look at like the specific schedule of medications and talked with your doctor about, how you might move some of those around to see if it impacts your RBD, that may be helpful. Yeah.
I apologize if I missed this question when I ran to the restroom. With RBD manifesting and already taking a certain amount of melatonin, is there a maximum, but after which it makes sense to try something else?
So melatonin, as many of you know, is available over the counter, right? Not a prescription a lot of times. Doses up to 15 milligrams, 1.5, have been studied for RBD in individuals with Parkinson's disease, but I think sometimes it's worth trying a different formulation of melatonin. There's also a prescription sort of version of melatonin that you can also try. And then, if you if you've sort of I think exhausted those options, been to higher doses, tried the long-acting prescription version, then that's when maybe a low-dose clonazepam might be necessary if the symptoms are very disruptive and frequent.
Thank you so much,
all.
Dr. Keener.
Yeah.
We hope you've enjoyed today's program. We're so glad you joined us. Special thanks to our amazing speakers, to Emily and Mackenzie from Reactive Physical Therapy. Were they great or what? Huh? It feels good to get up and move. If you enjoyed today, please make your tax-deductible donation by scanning the QR code or by visiting PCLA.org and make your donation so we can see you back again here next year. If you'd like to record a quick video testimonial for PCLA, for our social media, I'm going to be out in the front with the camera, and I'll help you through that. We'd love to hear from you. You can help spread the word and let others know about PCLA. Thanks again to our presenters today. And thanks again to our wonderful partners, Abbott, Acadia, Boston Scientific, Medtronic, and Supernus, and all of you attending. You make the events like this possible. PCLA wishes you a safe, happy, and joyful holiday season. See you soon.